Hi guys, am I audible, visible? Hi people, am I audible? Am I visible? Hi guys, am I audible? Am I visible? Hello. Happy morning guys, happy morning. So hi everyone, am I perfectly audible and visible? Am I perfectly audible and visible? Hi Hetwi, hi everyone, what's up? I think after the class, right, after this re-NEET examination, है ना, then you are going to miss this thing कि madam is late, yeah, she started session at this time or that time. What's up people? I am visible yaar, I am visible. Very good morning to all of you. I hope you all are doing good. I know you are excited, nervous, right? Mixed emotions are there. And finally, tomorrow you guys are having your exam. Trust me, I know this journey was just like a roller coaster. Okay? But finally, you guys will have your exam tomorrow. So make sure you attempt it properly because it is very important the way you attempt a paper. Okay, so today I'll post a short video for that too, that how to attempt the paper. So take care of that. And moreover, do carry your stuff properly, right? Your admit card, your ID proofs, passport size photograph, postcard size photograph. Along with that, yes, you can carry a transparent pouch, right? Just to avoid rainfall and all, right? Just to keep that admit card and ID proofs intact. Baki beta, you know that you're not allowed to wear any jewelry or something like that, so avoid it. And girls, you please put a simple rubber band, or even they can ask you to remove that. Do not put any clip or something. Okay, so these are the things that you have to take care. Okay, so these are the things that you have to take care. These are the things that you have to manage. Okay students, and if there is any confusion right after the class, you can put that in the comment section. So let's start the paper. Okay, let's start the paper. So beta, today the plan is very simple. Just you know that yesterday, right, we had this anti-authorized mock test. Meanwhile, you got the short questions as well. So this is what you people can see, right? Do check out this paper beta. In this particular paper, I have picked up the tricky important questions, and I've shared the PDF also. So today I will continue the same. And now, without speaking too much, otherwise you will be like, madam bus. So I am showing you your first question, right? I am showing you your first question. So here we have our first question, guys. So 1 2 3, go for it. 1 2 3, go for it. This is your first question, and I'm pretty sure today you are going to get all correct. Okay, today you are going to get all correct. So go for it. Beta, you will get good result, trust me, right? If you people trust me, then thank you so much, right? I am really grateful for that. And we have completed your class 11th revision like never before, class 12th with the help of shorts, and even before that also I took the individual, right, this NCERT reading sessions as well. PYQ practices there individually for plant physiology, human physiology, genetics, and even last 10 years PYQs, right? All your re-exams, be it in 2015, 2016, your 2022, 2024, right? All the re-exams also I have discussed with all of you. So guys, if you have followed all the lectures, trust me, you'll get good score. And not even all the lectures, even if you have completed that class 11th, class 12th marathon and these PYQ sessions, beta, you are definitely going to get good result. That is a promise from my side. Right? I rarely use this word promise, but yeah, that is a promise from my side. And today's session is also very important. I have picked up all the questions from today. Deliberately, I have picked up questions from the previous year papers only. Right? Today, you are having the questions from the previous year papers only, or tomorrow also in the morning, I'm going to meet you. There also, I will pick up the questions from the previous year paper, beta. Right? So you can trust me. There is no doubt in that. And let's discuss it now. Now, Alexander von Humboldt described for the first time, you know that he told us about the species area relationship. Now, how to revise it? You know that S is equals to CAZ. So you should know what is the meaning of this, and this, right? So C is the correct answer. I hope you all have marked it correctly. Excellent. Now, let's move to the next question. In case of poriferans, I'm starting with the simple one. Slowly, I will raise my level, right? Okay, and guys, mark your attendance by hitting the like button. How can you stop that? Mark your attendance by hitting the like button. People, quick. People, quick. Mark your attendance by hitting the like button, guys. Quickly. Excellent, excellent, excellent. In case of poriferans, the spongocoel. So should I increase the speed? If it is a yes, would like to see some fire emojis in the chat section. Should I increase the speed? Now, in case of poriferan, the spongocoel is lined with flagellated cells called choanocytes, right? Called choanocytes. And if you have practiced all the questions, you can also practice last 10 years PYQs. You can check that. Next question. Next question. Which among these is the correct combination of aquatic mammals? Focus on the word, beta. Aquatic plus mammals is the word, right? And if you want me to increase the speed, you can just send me some fire. Fire is there. Now, you should also increase your accuracy. You should also increase your accuracy. Aquatic mammals, right, beta? Cross, cross, cross. So, we are left with C. We are left with C. Whales, dolphins, seals. Am I right? Let's move to the next part. Okay. Trigon, superclass, Pisces. Next, which of the following represents the order of horse? This is again the PYQs, bacha. This is again the PYQs. My today's session is completely based on PYQs. Today at 10 p.m. the session that I'm going to take most predicted paper. That is again from, it is going to be the amalgam. New question, PYQs, statement-based question, few assertion and reason. I have designed that. Today at 10 p.m. also we are going to meet, beta, and we are going to solve it, and that too for 1 hour, right? In 1 hour we are going to solve all the 90 questions, because I will not take your much time. Or morning maybe we will just have PYQ practice only. Okay, PYQ practice. Now, which of the following represents? Now, beta, the order is, right? They are asking the order. So, first of all, these are to cancel. Equidae is the family. So, they are asking the order. So, it is Perissodactyla. So, even for the previous attempt also, I have solved these questions with all of you live. Oh, dekha. This is the mistake that you make. What are they asking, beta? What are they asking? They are asking us, right, guys? What are they asking? They are asking us the order. And what are you marking? You are marking the family. Isn't it? Isn't it? Isn't it? So this is the mistake that you have to avoid. I know the name for this modern horse is also Equus, right? It is also your Equus. Spelling mistake is there in Equus, but it is Equidae is the family. Perissodactyla is the order. You can check NCERT based MCQs. It is like that. Now, tell me a question from the male frog. This is the expected one. This is the expected one. Okay, you know that from frog either one or rarely two questions you people can get. So this is the expected one, and I really hope that you give the answer correctly. Otherwise, you will be punished. Yup. Everybody? Yes, beta? So it is going to be D, testis, vasa efferentia, kidney, Bidder's canal. In the male, you are going to see that urinogenital duct. And finally, there is the cloaca. So what is the cloaca? Cloaca is the common chamber. Common chamber where your digestive system opening, your excretory system opening, your reproductive system opening is going to open. So that's what you have to keep in your mind. Next question, beta. Which of the following is related to ex-situ conservation? So please attempt it properly. The word here is ex-situ conservation. The word here is ex-situ conservation. So ex-situ mane what? On-site, off-site. Ex-situ mane what? On-site, off-site. Again, off-site. So you are not going to protect that particular wild animal or plant in their natural habitat. Rather, you will move them to their special habitat. So what should be the answer here? Ex-situ. Dekho beta, this wildlife safari, it is ex-situ. Right, this wildlife safari is ex-situ. But if they will write, this can be confusing for some, if they will write wildlife sanctuary, right, then it is in situ. If you ever get confused, wildlife safari or wildlife sanctuary, what you have to do? Wildlife sanctuary, no, N is there in it. So it is going to be in situ. That's how I remember, right? It is going to be in situ. Aise to I remember it without that too. Chalo. An important characteristic that hemichordates share with chordates. Hemichordates share with chordates. An important characteristics that hemichordates share with chordates. The word is what? Hemichordates share with chordates. Quick. Hemichordates, you know that they are believed to be the connecting link in between the chordates and the non-chordates. So, what do you see here in the, what do you see? Matlab they are sharing. Absence of notochord, not at all. Chordates will have notochord. Ventral tubular nerve cord, the chordates are having dorsal, right? Dorsal hollow nerve cord. Pharynx without, no, beta, it is going to be with gill slits. So, pharynx with gill slit, या pharyngeal gill slits is the characteristic feature in the case of chordates. So, again, the answer is not up to the mark. They are saying which characteristic they share, beta. Which characteristic they share. So, this kind of mistake is not acceptable. Slap yourself. Chalo, slap yourself. Now, next part, the region of Biosphere Reserve, which is legally protected and where no human activity is allowed. Where no human activity is allowed. Right? Where no human activity is allowed. Yup. Yes, beta. Hanji, this is very simple. No human activity is allowed. So we know it very well. This is going to be core zone. So in Biosphere Reserve, what you people will see? You people are going to see the core zone, the transition zone, है ना? So there are transition zone, core zone or the manipulation zone. Which zone, sorry, manipulation or transition is same. So core zone is there, right? Transition zone is there, right? Transition zone is there and you are having buffer zone. So core zone mein there is no activity that they are allowing here. Buffer zone mein something related to research or like that. Transition zone mein yes beta there are management like locals, right? Scientists, they all are there in cooperation, right? They all are there. But yeah, here they have to make sure it should not harm the wildlife there. That is also the case. But in the case of core zone, no human activity is allowed. Next, viroids differ from viruses in. Like I think in last 2 or 3 days, we have solved this question thrice. So viroids differ from viruses. Who gave viroids? Mr. T.O. Diner. Which year, beta? Which year? 1971. Which year? 1971. Ma'am, section time. Ma'am, section time. Which section time, beta? I didn't get you. क्या नहीं समझ में आ रहा है बेटा? क्या रिपीट करूं? I didn't get this thing. Section time. I quick. चलिए. So viroids or viruses. Viroids बेटा, it is the infectious RNA. It is the infectious RNA. Right? And when it comes to the viruses, it is a nucleoprotein complex. Means it will contain nucleic acid plus protein, capsid, protein coat that is going to cover it. The nucleic acid can be DNA, it can be RNA. Right? The capsid is the protein coat which is going to cover it, which is going to protect it. Now, viroids where only infectious RNA is there, having low molecular weight, right? No protein part is there, no capsid is there. So that's what you remember. You have to remember. So RNA molecules without protein coat will be there. RNA molecules without protein coat will be there. Let's move to the next part. Mycorrhizae are the example of. We know it very well. Mycorrhiza, it is the symbiosis mutualism. So what do you see here? Roots of higher plants plus fungal hyphae. Roots of higher plants plus fungal hyphae. Let's move to the next question. An example of colonial algae. Very simple question. Direct question. Right. Repeated question. Direct question. Beta, if you want to get the poll, you have to download PyLens app. You have to download PyLens app. Okay? Otherwise, you can answer here in the chat section. That is also okay. Now, an example of colonial algae. The word is colonial algae. So this is going to be volvox. This is going to be volvox. So please answer such questions quickly. Spirogyra is filamentous, beta. Now this question, spliceosomes are not found in the cells of. The word is not found in the cells of. Beta, eco, human physiology, all the questions are done and dusted. Check last year, 10 years PYQs. Quick. 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 So spliceosome, where do you see? You are going to see them in the case of eukaryotes. You will see them in the case of eukaryotes. In the case of spliceosomes, you remember your snRNAs along with some proteins, right? So SNRPs are there. You will see. So snRNAs along with some proteins. SNRPs will be there. These small nuclear ribonucleoproteins. So they will be there in eukaryote. They are meant for splicing. They are meant for removing introns. So they will not be there in bacteria. So that is how you are going to remember that. That is how you are going to remember that. Let's move to the next question. Life cycle of ectocarpus and fucus. This is the exception. Don't be like ki life cycles are deleted from the syllabus. Still, many times when we discuss it, I have discussed it multiple times, beta. I have discussed it multiple times, beta. So quickly go for it. Quickly go for it. So life cycle of ectocarpus and fucus respectively are. What will you see here in the life cycle? In the life cycle of ectocarpus and fucus respectively are. So they are the exceptions, beta. Here you will see this is going to be haplodiplontic, and when you people will talk about the fucus, this is what? This is going to be diplontic. This is going to be diplontic. So C is going to be the correct answer here. And one more thing that you have to remember, both are the examples of brown algae. Right, your answers are clearly showing that you don't practice PYQ, and fucus is the algae having air bladders. Right, fucus is the algae having what? It is having air bladders. Right, fucus is the algae having what? It is having air bladders. Let's move to the next question. Smallest living cell, beta, simplest question. Smallest living cell without a definite cell wall, pathogenic to plant, as well as can survive without oxygen. Now understand the question here. They are saying it can survive without oxygen. It can survive without oxygen. Means normally it is living in the aerobic conditions, but it can survive without oxygen. So we also call them as, such organisms are also known as facultative anaerobes. Such organisms are also known as facultative anaerobes. Mycoplasma is the example. So they do not have proper cell shape. So it is pleomorphic, right? Because cell wall is not there, cell shape is not defined. So pleomorphic is the word, means having different shapes. Do you know that that is the reason they are also known as jokers of microbiological park या jokers of plant kingdom? मतलब you can also call them as jokers of, right, plant kingdom या फिर jokers of microbiological park, right? Jokers of microbiological park. This is also the term that you people can use here. Let's move to the next question. DNA replication in bacteria, it occurs. Let me see your concepts here too. Let me see your concepts here too. Yes, everybody, go for it. Go for it. All the related questions also I have discussed. Akhilesh, everybody here. So quickly answer it. And guys, hit the like button, mark your attendance too. Quick, go for it. So DNA replication in bacteria. So yes, you are right. Before, right? Not after transcription, not within nucleolus, not during S phase. Bacteria having no nucleus, nucleoid is there. So prior mane before fission, before this binary fission, this will be there. So C is the answer. Dekha? Again the common mistake. Again the common mistake. Ye dekho, 55% are saying S phase. Beta, S phase, eukaryotes. Chalo, let's move to the next question. Which of the following is not polymeric? Simple, lipids. Reference to the enzyme. Multiple times we have discussed that. So hollow enzyme will be coenzyme plus cofactor. So this is the example of complex enzymes which are made up, oh sorry, my bad, I am bad, sorry. So hollow enzyme is basically the complex enzyme. So it is made up of two things. That is the protein part, which is going to be apoenzyme, and then there will be cofactor. So cofactor is what? Cofactor is the non-protein part, beta. Cofactor is what? Cofactor is going to be the non-protein part, beta. And here in this cofactor, what you people are going to see in this cofactor, so it is having coenzyme, which is loosely bound, beta. It is having prosthetic group as well. It is having prosthetic group as well, which is tightly bound, and you will also discuss the metal ions there. You will also discuss the metal ions there. So B is going to be the correct answer. Now, a gene whose expression help to identify the transformed cell. We know that that is going to be selectable marker. What is it? That is going to be selectable marker. Chaliye na answer this. Association of histone H1 with nucleosome indicates what? Association of histone H1 with nucleosome indicates what? Quick, guys, quick. The association of histones H1 with nucleosome indicates what? With nucleosome indicates what? So you remember that I am explaining you the concept as well. So which histones do we have here? We talk about H1, H2A, H2B, H3, H4. है ना? H1, H2A, H3, H4. This is what we discuss if you remember. Now, your H2A, your H2A, H2B, H3, H4, they will be 2 copies of each is going to do what? 2 copies of each will make the octamer. Then your DNA will coil over it. So your H1 is basically the linker histone proteins. So when this H1 is associated here, so you will see that the DNA is condensed in the form of what? In the form of chromatin fiber. So that is what you have to see. So here nucleosome will have around 200 base pairs. So the logic behind I have already explained. So yesterday we took a session in the NTA authorized mock test where student was confused regarding what? Regarding the nucleosome number. So keep it in your mind, guys. Now this is the repeated question. Repeated question. How in the electrophoresis ethidium bromide will be used, bright orange colored bands will be there. Repeat it with me. Ethidium bromide will be used, bright orange colored bands will be there. Ethidium bromide is used, bright orange colored bands will be there. Let's move to the next part, guys. Let's move to the next part. So again, regarding this agarose gel, what is the criteria for DNA fragment movement on agarose gel during gel electrophoresis? What is the criteria for DNA, right? What is the criteria for DNA fragment movements on agarose gel during gel electrophoresis? During gel electrophoresis. Guys, speed up. During gel electrophoresis. Yup. During gel electrophoresis. Hanji. Yes. Smaller the fragment, the farther it moves. So always remember here, your anode is positive. DNA is negatively charged. Anode is positive. So your DNA after right staining under the influence of electricity as per size will move towards the anode. So the smaller the fragment size, the farther it moves. Next, the process of separation and purification of expressed protein before marketing. Before marketing. Yes, beta. So what will be there? There will be downstream processing. There will be downstream processing. Let's move to the next part. Hepatic portal vein. Right? I have discussed it multiple times. Hepatic portal veins. So they are going to drain the blood to the liver from. They are going to drain the blood to the liver from. Blood to the liver from. To the liver from. From where? Hanji, exactly. So it is going to be the intestine. So it will not, that blood from intestine, deoxygenated blood from this intestine will not directly pass to the heart. First, it will go to the liver. So this is the hepatic portal vein. So we have hepatic portal vein. You will also see that in the case of frog. Frog mein to you will also talk about renal portal vein. Renal portal system. Now, next part. Answer this. Answer this. In the case of, yes, everybody, adult human RBCs are enucleated. Guys, speed up. Adult human RBCs are enucleated, right? Means they are without nucleus. What is the most appropriate explanation for that? And I'm sure you guys can answer. I'm sure you guys can answer. Like the most valid explanation. So they do need to reproduce. They are somatic cells. They do not metabolize. All living cells, they do metabolize. All living cells, they do metabolize. So all their internal space is available for oxygen transport. So here A is going to be the correct answer, beta. A is going to be the correct answer. Now, MALT. What is MALT? MALT stands for mucosa associated lymphoid tissue. It stands for what? Mucosa associated lymphoid tissue. Mucosa associated lymphoid tissue. So it makes 50%. 50% of the lymphoid tissue in human body. Right, beta? Now, next question. Speed up, everybody. Lungs are made up of, right, beta? Lungs are made up of air-filled sacs. The alveoli, they do not collapse even after forceful expiration. Forcibly you are expelling, right? Right, then still there is no, right? It is not even collapsing. What could be the reason? What could be the reason? What could be the reason? Exactly, it is going to be residual volume. It is going to be residual volume. So it is not going to be always empty. Some amount of air will always be there. Chaliye, let's move to the next question. Attractant and rewards are required for, beta, they are required for entomophily. What is entomophily? Pollination by insect. What is anemophily? Pollination by wind, right? Hydrophily, water. And when you are talking about cleistogamy, you are talking about the closed flower, where definitely autogamy will be there. So here, right, beta, what is the topic that you, what is the answer that you have to remember? Attractant and rewards are required for entomophily. Now, which of the following component is going to provide sticky character? Sticky character is my keyword here. So that is going to be your glycocalyx. That is going to be your glycocalyx. Next question, beta. Chaliye, answer this question. Which of the following options give the correct sequence of events during mitosis? Yup. Go for it. Everybody go for it. Everybody go for it. Shabash. I can see there are few spammers only, and today our serious NEET aspirants, they are not letting them, right? They are not letting them say anything. Right? They are not letting them do anything. There are few spammers. But today my students, they are clear. So beta, first of all, the thing that you have to see, right? The thing that you have to see here is ki what are they asking exactly? They are talking about the mitosis. So if it is mitosis, crossing over will definitely not be there, right? Here also crossing over will not be there. Now we are left with two options. Condensation, theek hai ji, nuclear membrane disassembly, arrangement at equator, centromere division, then segregation mane separation, then telophase. Condensation, arrangement, centromere, segregation. So out of that we can go for B, right? Properly it is explaining that. Okay, chaliye, zygotic meiosis. Beta, zygotic meiosis mane when zygote is undergoing meiosis, right? There are many organisms that will show that. In the algae also you are going to see, right? Because algae, it shows what? It shows haplontic life cycle. Haplontic life cycle. So haplontic life cycle mein main plant body is going to be haploid. So your zygote, right, which will undergo meiosis. Zygote is the one that will undergo meiosis. And when zygote undergoes meiosis, beta, we can also call it as zygospore. So there is a difference in zoospore and zygospore, right? There is a difference in zoospore or zygospore, right, beta? So chlamydomonas is going to be the answer here, because in the case of, right, Markensia, firstly mitosis will be there, fucus is showing diplontic life cycle. So the answer here is going to be chlamydomonas. Next part, beta, anaphase promoting complex. It's a PYQ again. It's a PYQ again. Very good, everybody, speed up, right? And students today, do not let these spammers spam, okay? Do not let these spammers spam today, right? Let them say whatever they want to say, right? Arey madam, why are you not teaching in Hindi? Yet this and that. Let them say whatever they want to say, but you make sure you are not going to let them spam. Okay? You maintain the pace. You maintain the pace. You spam the answers here. Now they are saying APC. What is APC? What is APC? Anaphase promoting complex. It's a protein degradation machinery necessary for proper mitosis if APC is defective. They have already explained you the meaning here. So if APC is defected beta in the human cell, so what will not happen? APC, anaphase promoting complex, promoting anaphase, which will segregate the chromosome. So obviously beta, chromosomes will not segregate, isn't it? Chromosomes will not segregate, isn't it? Chaliye, let's move to the next question. A decrease in blood pressure will not cause the release of Mr. ANF, natriuretic factor, and do remember it's a vasodilator. So already when the blood pressure is low, how that vasodilator will release? Yeah, how this ANF will release? It will not release. Next question. GnRH, a hypothalamic hormone needed in the reproduction, it acts on. Very simple, it is GnRH. I hope you know the full form. I hope you know the full form. Gonadotropin releasing hormone. What is the full form that we have here? Gonadotropin releasing hormone. Am I right? Gonadotropin releasing hormone. Am I right? Am I right? Hanji everyone. Yes, beta. So they are saying gonadotropin releasing hormone. So we know that GnRH, so anterior pituitary and stimulates LH and FSH, right beta? So anterior pituitary, right? So you know that pituitary is having two parts, adenohypophysis, neurohypophysis. When you people talk about adenohypophysis, you will also see there will be anterior lobe referred to as pars distalis. There will be intermediate or middle lobe, which is known as pars intermedia. So that is something we need to keep in our mind. So B is going to be our correct answer. Let's move to the next part. Hyposecretion of growth hormones. If growth hormones are excess in adult, what is going to happen beta? What is going to happen beta? Beta, I will speed up. Definitely I will speed up. So hypersecretion of growth hormone in adults does not cause further increase in height. Why there is no increase in height further? So obviously we know that epiphyseal plates, they will close after the adolescence, right? So because of that, further height will not increase. Is that clear? Further height will not increase. Is that clear? Is that clear? Thank you so much, beta. Thank you so much. So this is going to be our correct answer, right? Next question, a temporary endocrine gland. Temporary endocrine gland, can we call it as corpus luteum, the yellowish body going to release progesterone, right? It is the temporary endocrine gland. Chali, next part. Which ecosystem has highest biomass? My students are going to answer it in less than 30 seconds. I'm sure about it. I am sure about it. You are going to answer it in less than 30 seconds. I'm sure about it. So go for it, beta. Go for it. So which ecosystem has maximum biomass? Maximum biomass, organic matter, biomass, maximum. Obviously, beta, it is going to be your forest ecosystem. What is it going to be? Excellent, beta. It is going to be your forest ecosystem. Next question. Some students are still marking grassland. Beta, you please see the question. Now, presence of plants arranged into well-defined vertical layers. Beta, this question I have repeated thrice, even in my short shorts for that class 12th, right? I hope you guys have seen that short short for class 12th. So in that, vertical layer is the word. So beta, this is the stratification that you are talking. So where will you see that? Now they are saying presence of plants into a well-defined vertical layer. Vertical layer mane, you are talking about the stratification here, right? You are talking about the stratification here. Quick, everybody. You are talking about the stratification here. Yes or no? Yes or no? So if there is a stratification, vertical distribution is there. So where are we going to see that? Where are we going to see that? Obviously, we are going to see that in the tropical rainforest. We are going to see that in the tropical rainforest. Next question, sewage treatment will remove the suspended solid. Obviously, that is going to be the primary treatment. Remember, filtration, sequential filtration and then sedimentation. Sequential filtration and then sedimentation. Chaliye, let's move to the next question. Which of the following is correct? Which of the following is correct? I believe you are going to answer it correctly. Which of the following statement is correct? We will discuss it also. Ascending limb of loop of Henle. Okay, Ji, this is descending limb. This is going to be ascending limb. So ascending limb of loop of Henle is impermeable to water. Yup. Descending limb is impermeable to water. No, beta. Why? Because it is permeable to water. Right. The ascending limb of loop of Henle is permeable to water. No, beta. Why? Because it is impermeable to water. And descending limb is permeable. No, it is going to be impermeable. From the ascending limb, you know that electrolytes will come out. So which one is correct? A is correct. Right. Which one is correct? A is correct. Still, 70% students were able to give the correct answer. Not good. Now, transplantation of tissue organ due to non-acceptance by patient's body. So, immune response and such type of rejection will always be from your CMIS. So, what is your CMIS, cell-mediated immune response? Here, your T cells are participating. Who's participating here? T cells. Beta, we will also have statement-based question. Right now, so you are having direct questions. We will also go for statement-based questions. So now, root hairs develop from the region of maturation. Right? Root hairs are having a fixed job. They will arise from the maturation region. Now, which of the following will facilitate, give it a try, which of the following will facilitate the opening of stomatal aperture? You cannot say that it is from the deleted portion because the part of it we have also covered, and this is something that you should know. And I'm sure you will answer it. You are good people. You are amazing people. Yes. So, stomatal aperture. Dekho beta, in the case of stomata, always remember your guard cell. Inner walls are going to be thick. Outer walls are thin. And then you have parallelly arranged cellulose microfibrils, which are radially oriented. Now, contraction. And here, turgidity of the guard cell is the one that will decide that opening and closing. So, radial orientation, decrease in turgidity. They are saying opening to beta, the cell should be turgid. So radial orientation. So this is the correct answer. Radial orientation of cellulose microfibrils in the cell wall of guard cell, not the fourth one. Now capacitation, it will be there in the female reproductive tract. What is the role of this capacitation? The charging of sperm. If the sperm will not be charged, it will not be able to fertilize. It will be trapped in cumulus oophorus. So in capacitation, calcium ions are also very important. The the cholesterol, even the structures which are covering the sperm surface receptor, they will be removed in this charging. Very important it is. Next part, again a very simple question. I'm pretty sure you guys will answer it correctly. Now go for it. Again, it is from the PYQs. Again, it is from the repeated topics. So go for it, everybody. Yes, I'm sure you can answer it. I am sure you can answer it. Yes, beta. So it is very simple. They are saying X pair of ribs. How many pair of ribs do we have? 12 pairs. 12 pairs of rib. Only 7 pairs are. So word is pair. So accordingly, you have to go for it. So what is going to be the correct answer? Sorry, 12 pairs. Hanji. So X is 12. 12 pairs. So 12 pairs is equals to total 24 ribs are there, right? Right. So let me see the option out of dash pairs. Where is it? X is, acha, here. Okay, my bad. X is 12 pairs, and Y is 7 pairs. So they have just shuffled it. So true ribs are attached dorsally to vertebral column, ventrally to sternum. Very good. So A is going to be the correct answer. Chaliye. Next question. Again, go for the next question. I'm sure you are not going to make any mistake here. And guys, see, you have worked hard. You are going to give your paper and you are going to enter in that examination hall like, yeah, you know everything, right? And whatever is going to happen, there should be no regret. Just give your best. That should be the attitude. That should be the attitude. Okay. Okay. Chaliye, go for it. Go for it. So now the next thing is, if there are triple line bases, you know that 3 bases will give you 1 amino acid, right? So there will be 333 amino acids. Why? 999 bases are there, right? 333 amino acids are there, and 901 at this position the base is altered, so the length of RNA will become 998. So how many codons will be altered there? Then obviously beta 33 codons will be altered, because up to 900 things are fine, so 300 are perfectly okay, so 33 will be altered. So this is something I have discussed multiple times in my classes. Chaliye, final proof for the genetic that DNA is the genetic material. Hershey and Chase. Hershey and Chase. Transduction experiment. Next question, bacha. Which of the following RNA should be most abundant in the animal cell? Most abundant RNA. Most abundant RNA. So when it comes to most abundant RNA, this is going to be your ribosomal RNA. Ribosomal RNA, and least abundant is your mRNA. Do remember that. Least abundant is your mRNA, most abundant is your ribosomal RNA. That is something that you all need to remember. Am I clear? That is something that you all need to remember. So rRNA, and here this is going to be the least abundant one. Am I clear? Am I clear, everybody? Am I clear? Sure? So, most abundant rRNA. Now, during replication, Okazaki, beta, I will again explain you. Okazaki fragments, they are the fragments, they are making the lagging strand. They are growing away from the replication fork, right? So, this thing you have to consider, ki lagging strand is there, but they are growing away from the replication fork, right? They are growing away from the replication fork. Leading strand is growing towards the replication fork. Lagging strand is growing away from the replication fork. Let's suppose this is my double-stranded DNA. Let's suppose this is my double-stranded DNA. So here what do I have? This template strand is having polarity 3' to 5'. This template strand is having polarity 5' to 3'. So the template strand having polarity 3' to 5'. Obviously, initially you need RNA primer, and then what is going to happen? You are going to add the bases. So it is towards the replication fork. Now, when I talk about this particular part, right, beta, this particular part. So here you people will see that lagging strand will grow away from the replication fork. So this is what you have to remember. So this is the lagging strand. This is the lagging strand. Is that clear? Is that clear? Right? So again, next question, in Bougainvillea, thorns are the modified. Beta, this is something important. This is something important. So in Bougainvillea, thorns are the modifications of. Thorns are the modifications of what? In Bougainvillea, thorns are the modifications of what? In Bougainvillea, thorns are the modification of what? Hanji, thorns are the modifications of what? So we know it very well. They are the stem modifications, right? Stem modifications. Next question. Next question. Morphological nature of edible part of coconut. Nothing but the endosperm. Next question. Functional megaspore develops into. Functional megaspore will develops into embryo sac. So embryo sac is going to be the female gametophyte. What's that? Female gametophyte. So coconut fruit is what? It's drupe. One word answers. Let's move to the next part. Chaliye. Again question is from frog. Again question is from frog. Do remember this particular question. Do remember this particular question. Again it is the easy one. Right. Again it is the easy one. And I'm sure you all will answer it correctly. Right. I'm sure you all will answer it correctly. Guys, quick. Yes. So they are saying frog's heart when taken out of the body will continue to beat for some time, right? Because this heart is also myogenic, it is auto-excitable, right? Heart is myogenic, heart is auto-excitable. Heart is myogenic, heart is auto-excitable. Is that clear? Is that clear? Beta, see, heart is, because myogenic, right, muscular heart, it is having that patch of muscles which will excite the wave of, initiate the wave of contraction. So outside the body also it is going to be auto-excitable. Now, myelin sheaths are produced by, I'm sure you guys can answer the question. Myelin sheaths are produced by, yes, beta. Myelin sheaths are produced by, so we know that Schwann cells and oligodendrocytes, they are going to make it. They are going to make it. So Schwann cells, where do you see? Right, we see that in PNS. This is oligodendrocytes we will see in the CNS, central nervous system, and you are having the peripheral nervous system. Central nervous system, and you are having the peripheral nervous system here. Next part, receptor sites for neurotransmitters are present on the, receptor site for neurotransmitters are present on the, right? Feeling confident, guys? Feeling confident? So receptor sites, where do you find the receptor sites? Where do you find the receptor sites? Hanji, so receptor sites for neurotransmitter, they are on the postsynaptic membrane. Postsynaptic membrane. So if you don't know, just say, let's suppose you are having a neuron, right? Thank you. This is, let's suppose you are having a neuron, so that neuron is having the nerve endings, so this is making the presynaptic neuron. There will be the gap in between, so there is next neuron, right? The dendrites of that next neuron and all. So this is what you people can see here, right, beta? So this is your postsynaptic neuron. In between there is the synaptic cleft. So obviously postsynaptic neuron is going to be the answer. Asymptote. What is asymptote? What is asymptote? Asymptote is K is equals to N. When carrying capacity and population size is equal, there is no growth beyond this point. Next point, beta, phosphoenol pyruvate. Phosphoenol pyruvate is the primary carbon dioxide acceptor in the, phosphoenol pyruvate is primary carbon dioxide acceptor in the, where will you see as the primary carbon dioxide acceptor? Right, beta, where will you see the primary carbon dioxide acceptor? Phosphoenol pyruvate. Phosphoenol pyruvate. Phosphoenol pyruvate. So phosphoenol pyruvate is a 3 carbon containing compound. In the case of C4 plants, you are going to see that, right? It is the CO2 acceptor in low C4 plants. Now you can see the options C3, C4, C2, C3, C1. So it is going to be C4 plants. It is going to be C4 plants. Phosphoenol pyruvate, beta. Phosphoenol pyruvate is not there in the C3 plants. It will be in the C4 plants. In C3 plants, you are having 5 carbon containing compound, that is your ribulose 1,5-bisphosphate. Ribulose 1,5-bisphosphate. Now, which statement is incorrect for the Krebs cycle? Which statement is incorrect for the Krebs cycle? I'm sure you guys can answer that. Which statement is incorrect for the Krebs cycle? Speed up, people. Which statement is incorrect for the Krebs cycle? Yes, beta. Hanji, which one is? So what are they asking us? Wrong. So there are 3 points where NAD is reduced to NADH to true. Do remember the reaction. 1 point where FeDH2 will form, true. Succinyl coenzyme to succinate a molecule of GTP synthesized, true. Cycle starts with the condensation of acetyl coenzyme with pyruvic acid, no beta, with oxaloacetate to yield the citric acid. So this is going to be D. This is for the third time we are solving the same question, right? Now beta, next question in reference to the factors, affecting the rate of photosynthesis, which one is not correct, right? Which one is not correct? So they are saying ki with reference to the factors affecting rate of photosynthesis. So one question for this particular year you people can expect. You people can expect one question, right? You people can expect one question this time from this part. Beta, light saturation, they are saying which is not correct. Light saturation for CO2 at 10% of the full sunlight. Increasing atmospheric CO2 up to 0.05 can enhance CO2 fixation rate. C3 plant respond to higher, no beta. C4 plants have much lower, no beta. So C3, this C is incorrect. Baki tomato is a greenhouse crop which can be grown in CO2 enriched atmosphere. That is absolutely correct, beta. Chaliye, let's move to the next part. Fruit and leaf drop. Fruit and leaf drop, right? You can prevent by the application of auxin. Select the mismatch. Okay, go for it. Select the mismatch. So these examples can be confusing, right? So quickly answer it. So select the mismatch, guys. Select the mismatch. Yes, everyone, select the mismatch. Beta, it is simple, right? Your pinus is not going to be dioecious, it is monoecious. It is bisexual. Salvinia is heterosporous, Cycas is dioecious, Equisetum is homosporous. So A is going to be the correct answer. Next question, plants which produce characteristic nematophores and show vivipary. Again, we have solved it earlier, so it is going to be helophytes. Now, again select the mismatch. Right, beta, right. Again select the mismatch. Quick, go for it. Yup. Yes, it's very simple. They all are correct, beta. Frankia alnus, right? Anabina, nitrogen fixer. Rhizobium, alfalfa. This is incorrect, beta. Mycorrhiza, you know what exactly mycorrhiza is. And this rhodospirillum is anaerobic, beta. It is photosynthetic and even it is the nitrogen fixer, right? So this bacteria. So this is what you have to remember. B is the incorrect answer. So disease caused by primary non-disjunction, but autosomal primary non-disjunction. Guys, speed up, right? The disease caused by an autosomal primary non-disjunction. Quick. Quick, everyone. Quick, everyone. Beta, the word here is autosomal. So if it is autosomal, it is going to be Down syndrome. So you will talk about trisomy 21, right? If they will ask the same thing, but in the case of sex chromosome, in the case of allosomes, the answer is going to vary. Chaliye, next part, thalassemia. This is again the same repeated question. Again, I'm telling you guys, please pay attention. Thalassemia, it is the this is the quantitative disorder. Sickle cell anemia is qualitative, because here that beta hemoglobin in that at 6th position amino acid will be changed. RBC will be sickle shaped. Thalassemia mein it is about the quantity, right? So thalassemia is due to less synthesis. Yes beta, this is the correct one. And sickle cell anemia, it is NCERT bacha, it is NCERT. So this is going to be your qualitative, this is going to be your qualitative disorder, and this one is going to be quantitative. This one is going to be what? It is going to be quantitative. That's what you have to remember. Chali, the genotype of husband and wife is given. The genotype of husband and wife is given. So now they are saying how many different genotypes and phenotypes are possible. Husband and wife, right? One is AB, another is heterozygous A. So obviously in that case, you can see that, and even you can make it, but this is simple that we can answer directly as well. So A blood group, homozygous A, heterozygous A. AB blood group is also there beta, and heterozygous B is also there. So here the phenotype, if I talk about, so 3 phenotypes are going to be there. But if I talk about genotype, 4 genotypes are going to be there. This time I'm not marking it incorrectly. So homozygous A, heterozygous A, this AB codominance, and then this. So C is going to be our correct answer. Chaliye, next question. Next question. Again, it is from STIs. Yes, the again question is from STIs. So go for it. Yup, go for it. Everybody just go for it. Yes, beta. Yup. So gonorrhea, Neisseria gonorrhoeae, syphilis, treponema pallidum, genital warts beta, human papilloma virus, and AIDS will go here. So A with 2, B with 3, C with 4, D with 1. So it is going to be A as the correct answer. Next part, which of the following is the function of copper releasing IUD? Copper releasing IUD, copper T, copper 7, multiload 375. What is going to be the function? Yes, exactly. So can we say that A is going to be the answer here? Can we say that A is going to be the answer here? So they will suppress sperm motility and fertilizing capacity of sperm. They will suppress sperm motility as well as fertilizing capacity of sperm. Next part beta, now they are saying there is a couple where the male is having low sperm count. Which technique? Male is having low sperm count, so artificial insemination will be preferred. Artificial insemination will be preferred. So insemination is the transfer of male gamete inside the female genital tract. So artificially it will be done. Now dioecious flower. Dioecious flower will prevent what? So you should know what is the meaning of dioecious flower. So do remember one trick of mine, that is BMH. What is BMH beta? BMH is bisexual, monoecious, hermaphrodite. BMH is bisexual, monoecious, hermaphrodite, right? Hermaphrodite means it is bisexual, single individual is having male and female. So naturally dioecious is going to be what? Dioecious is going to be unisexual. So if it is going to be a dioecious flower, so autogamy will not be there. Autogamy will definitely not be there. So it is going to prevent beta. It is going to prevent both. Both what? Autogamy, gitonogamy? Nahi. So it will prevent dioecious माने unisexual. So it will prevent xenogamy nahi, that is the cross one. So out of all right? Out of all the options that are available to us, we will go for B. Why? Right? Autogamy will not be there, but beta gitonogamy, but it is the flowering plant, the complete plant actually. The complete plant actually is unisexual, so neither autogamy nor gitonogamy will be there. Now in gitonogamy, you will be having monoecious plant. In gitonogamy, the plant is going to be monoecious, right? And flowers are what? Flowers are going to be unisexual, flowers are going to be dioecious. So this is the condition that you have to remember. So in this gitonogamy, what is going to happen beta in gitonogamy? In gitonogamy, you will see the plant is going to be monoecious, but the flower is going to be dioecious. Mane, you will be having the same plant with male and female flowers. So let's suppose if I'm denoting it, this is going to be female, this is going to be male flower. So in that case, gitonogamy will be there. So basically the gametes will transfer. The flower is different, but the plant is same. So genetically, no change will be there. So castor and maize, coconut, cucurbit, these are the examples. Okay, these are the examples. Next, move to the next question. So flower which have single ovule in the ovary. Ovule is single in the ovary and packed into inflorescence and pollinated by. Packed into the inflorescence are pollinated by. I am sure you can answer it. I am sure you can answer it. Yes, everyone, I'm sure you people can answer that. Hanji, everybody would like to see the answer here in the chat. Very good. So it is going to be wind pollination. Next, double fertilization is exhibited by your angiosperms. Let me tell you, gymnosperms are also having endosperm. In the case of gymnosperms, definitely you people will see endosperm, but that is going to be haploid. Why beta? Because in gymnosperms, there is no double fertilization and this endosperm is also made from the female gametophyte. Okay, now, chalo, let it be. This is also the deleted part. I don't know why it is still there. Chalo, let's go for it. The probable reason for the grayish appearance of cerebral cortex. The probable reason for the grayish appearance of cerebral cortex. Yup. Why that cerebral cortex is grayish? So it is going to be the cell bodies of neurons are concentrated here. That's why beta it is having the grayish appearance. Matlab, how can you mark all of above? They are contradictory as well. Presence of bundles of axon. Yeah, cell body of neuron is having. No, the cerebral cortex is grayish in appearance. Why? Because of that, right, this cell bodies are concentrated there. Next question, select the incorrect for the vexillary or the papilionaceous corolla. Again, the important part. Again, the important part. Everybody speed up. Again, the important part, everybody speed up. So they are talking about what? Papilionaceous corolla, vexillary aestivation. Remember, you'll be having one largest petal standard or vexillum on the posterior side, right? Wings or alae. Then they have what? They are having wings or alae. And then they are having what? Then they are having the keel or carina, which are going to be fused. So it will be 1 plus 2 will be fused there. Is that clear? Plus 2 will be fused there. So two lateral petals are found which are forming wings. Standard petal is posterior. That is also correct. Larger petal is anterior. So two smallest anterior petals are also there. So yes, beta, this will be at the posterior side. I have discussed it earlier as well. This is going to be at the anterior side. So this is keel or carina and they are fused and they are asking incorrect statement from here. So you can see majorly people are marking C, right? The largest petal is on anterior. Nahi beta, the largest petal which is known as standard or vexillum, the largest petal which is known as standard or vexillum beta, it is on the posterior side, it is not on the anterior side. Got it beta? It is not on the anterior side, it is on the posterior sides. Are we clear here? Are we clear? Sure? Sure? Right? So D is the correct one only and please do not repeat this mistake. Okay, chaliye, this one, epipetalous stamen. Epipetalous condition means what? Your this androecium and these petals, they are fused, and mainly in the case of Solanaceae family, you are going to see that character, right? Mainly in the Solanaceae family, you are going to see the character. Hanji beta. So where do you see that? We have discussed this again, right? Do you remember the answer here? Just say your tobacco, tomato, your chili, है ना? So one more example, this petunia, they all are the members of Solanaceae, which will show that. So this is going to be the answer here, right? It is not 6, it is D, right? It is 4. नहीं? चलिए, let's move to the next part. Which pair is not having similar type of roots? Which pair is not having similar type of roots? Which pair is not having similar types of roots? Guys, quick. Yes. So look at the question, read it carefully. They are saying not similar type of roots. Yes. देखो बेटा, here the answer is going to be mustard and maize, because one is dicot, another is monocot. So here you will see the tap root, right? And here you are guys are going to see the fibrous root. So it is going to be A. It is going to be A, right beta? Chaliye, let's move to the next question. What is incorrect about prothallus of pteridophyte? So what is the prothallus of pteridophyte? What is the prothallus of pteridophyte? So prothallus is going to be what? Prothallus is going to be gametophyte. Prothallus is going to be gametophyte. So prothallus of pteridophyte is having what? It is having the gametophyte beta. So they are so asking incorrect statement. Well differentiated with vascular tissue? No. Always remember, in plant kingdom, gametophytes are avascular. You can talk about any of the gametophyte. They are all avascular. This is a fact regarding plant kingdom. This is small and undifferentiated. This is the gametophytic phase. That's true. This is multicellular and free living. That is also true. So this prothallus, which is there in the pteridophytes beta, right? It is having that sex organs also. It is having the rhizoids also. It is small and photosynthetic as well. So B is going to be the correct answer. Why? Because they are asking the incorrect statement. So yes, it is the gametophytic phase. Beta, incorrect statement they are asking, right? So this is right. Oh, 1 second. 1 second, 1 second. Yes, A is the incorrect statement. My bad. This is small and undifferentiated. A is the correct answer. Beta, I'm sorry, I marked it incorrectly in the poll. A is the answer. Hanji. I'll give you the marks. But still, majorly students have marked C, C also. So this is the gametophytic phase, but they are not asking the true statement. Incorrect statement they are asking. So A is the answer. Why? Because this statement is incorrect. This is well differentiated with vascular tissue. No. In the plant kingdom, there is no gametophyte which is having the vascular tissue. So I know you will ask for justice. So you can answer it again. Answer it again. Quick. I am sorry, Babu. Chalo. हाँ जी, ठीक है. So free bonus marks, ये bonus marks. चलो. So A is going to be the answer here. A is going to be the answer here. चलिए, next part. Structure of human sperm is given, right? Structure of human sperm is given. Identify the part which possess enzyme that help in fertilization as well. Identify the part which possess enzyme and help in fertilization as well. Guys, quick. And help in fertilization as well. Quick. Quick. Identify the part. So don't you think it is going to be A, right? Head part where acrosome is there, and that will be having the enzymes which are required for the fertilization. Next part, which set of plants of gymnosperms grow in temperate areas with a branch stem, right? This is the hint here. Branch stem, needle like leaves to reduce the surface area. Matlab they can survive, or they should survive in that xeric conditions as well. And the word is gymnosperms, beta. And the word is gymnosperms. Even if you will go for the elimination method, you can clearly opt it right. Right? You can easily opt it right. You will easily opt it right. Yes. देखिए बेटा, see, eucalyptus is not gymnosperm, Wolfia not gymnosperm. They both are gymnosperm, but your cycas is unbranched. Here, Pinus and Cedrus is going to be the answer, because yes, you are right. They are going to be branched, beta. They are going to be branched. So it is not C. It is not C. Beta, they are saying branched stem. In the case of cycas, don't you remember unbranched stem? Right, at the top of it, you will be having the leaves. So in cycas, the stem is going to be unbranched. What sort of stem do we have? Unbranched stem. We also use the term caudex for it. Caudex is also the term that we can use for it. Chaliye, let's move to the next question. Which one is not property of ideal contraceptive? It should give beta. So B is going to be the answer, right? Which is not according to the binomial nomenclature. First word represent the genus, second represent species. Names are written in Latin and are italicized. Biological names can be written in any language, not at all. If the name is handwritten, it has to be underlined separately. Yes, beta. Right. Next question. Identify the feto-maternal organ. Again, very simple it is. They are saying identify the feto-maternal, right? Feto-maternal organ, that's the word, right? Identify what? Identify the feto-maternal organ. Feto-maternal organ. Yes, bache. Yes, beta. So, obviously it is going to be placenta. So, fetus, chorionic villi. है ना, from fetus side chorionic villi will participate here. From mother's side, that endometrial part. So C is going to be the correct answer. Now the next question. Answer this question. Now they are saying beta ER is also called. You have to find not true. Not true mane you have to find incorrect statement. So while attempting the paper, please pay attention. Please go. Please focus on such keywords, beta. Right? Please focus on such keywords. Yes. They are saying, right, which one is not correct? That's the question. What's the question, beta? The question is which one is not correct? देखिए बेटा, ER is the control center, not at all. Why? Because it is going to be the nucleus. It modifies the chemicals that are toxic to cell. Yes, that's the job of smooth endoplasmic reticulum. It is the major site for protein synthesis. No, beta, this is going to be the lipids. So this is also incorrect. Incorrect. It's a network of interconnected cisternae, tubules, and vesicles present in that endoplasm. So 1 and 3. Chali, next one. In which stage does the embryo differentiate into trophoblast and inner group of cells? Embryo differentiate into trophoblast and inner group of cells. So I hope you remember what are you talking here. You are basically talking here about the blastocyst. About the blastocyst, and here it will be called as blastula. Why blastocyst? Because it is having this cavity which we are calling as blastocele. So these are the trophoblast cells. So these trophoblast cells will release the proteolytic enzyme which help in their, right, which help in their implantation there. Then this part is what? This is the inner cell mass. This part is what? Inner cell mass. So it will form the future embryo. It will form the future embryo. Right, beta? Then here what do you have? This part is the cells of Robber, right, which are directly cells of trophoblast, which are directly in contact with this part. They are going to be cells of Robber. So C is going to be the answer here. Next part, again, match the column type of questions. Easy they are, so go for it. Go for it. Guys, speed up. Speed up. Yes. Oh, 1 second. Anaphase में sister chromatids are broken down. So B will go with 3. Eliminate these 2. Telophase में reformation of ER will be there. Right? C में there will be formation of opposite spindle, and D में spindle grows between the centrosome. So as per the options given here, B is going to be the correct answer. The enzyme which convert glucose to ethyl alcohol. Glucose to ethyl alcohol. This is going to be your zymase. This is going to be your zymase. Select the correct match here, beta. So alluroplast, proteins, tuber, acha nahi. Correct match. Okay, so yes, alluroplast. They'll store what? They'll store protein. Now you answer it, everybody. Quick. Chlorophyce appears grass green due to chlorophyll A and B, right? Due to chlorophyll A and B. Now I will be super duper fast. Now blood stem cells are related to which of the following options? Chali, give it a try. Give it a try. Sometimes simple questions can be confusing too. Yes, beta, I'll be too fast. Now please answer this. Now please answer this. In 15 seconds, I will end the poll. It's very simple one, but sometimes students, right, they make mistakes. So obviously it is going to be what? It is going to be bone marrow. It is going to be bone marrow. Next, statement 1, statement 2. Diplohaplontic or haplodiplontic is the characterized by existence of sporophyte as well as gametophyte. Fungi is having haplodiplontic. Nahi, हाँ, fungi is having haplodiplontic. Beta, fungi mein, it's not haplodiplontic, mainly fungi is going to be haploid. Now, AZT, it is useful in the treatment of this azidothiamidine. Yes, beta, this azidothiamidine. Let me tell you, in the case of HIV infection, right, people, they get it, but ultimately, what exactly it is antiviral or retroviral rather. Okay, beta? Right, so an RNA containing the viral borne disease. So this is going to be A, right? This is going to be A. If you know it, very good. Otherwise, do remember that. Next part, select the incorrect statement. Yes, everybody, select the incorrect statement. Now, again, I will be super duper fast. So you please answer it. You please answer this. They are asking incorrect. Is the hemoglobin example of quaternary protein? Yes. In the primary structure, the left hand will be beta N-terminal. Always remember CONH. First amino acids, carboxylic group will participate there, right? So this is what you have to remember. Right hand will be having the C-terminal. In proteins or polypeptides, the amino acids are linked by peptide bond. Most of the enzymes are protein in nature. So this is going to be B. This is going to be B. Now, which type of selection is right? Which type of natural selection in a population is rare in occurrence than others? Is rare in occurrence than others? Guys, quick. Which one is rare in occurrence than other? Yup. The word is rare here. The word is rare here. So it is going to be disruptive. It is going to be disruptive. If you know it, very good. If you don't know it, you should know it. Next part, haploid content. Haploid content will be 3.3 into 10 raised to power 9 base pair. Do not confuse it with 6 base pairs. Let's move to the next part. Malonate. Malonate. This year we have practiced this question so many times. It is inhibited by succinate dehydrogenase. Malonate is inhibited by succinate dehydrogenase. Basically, malonate is going to inhibit the reaction catalyzed by this enzyme. So this is the example of competitive inhibition. Okay, this is the example of competitive inhibition. Next question, beta. Tasmanian wolf, they resemble the placental wolf. This is the example of. This is the example of. They are so different, beta. So this is basically the example of. Quick. Quick. Hanji. Beta, see, these two are different from each other. So, but still, so they are representing what? Still they are representing what? Parallel evolution. Matlab, they are so, beta, how come divergent? Divergent may same ancestor, right? Ancestor is going to be same, but it is not like that here. Right, they are individually, they are showing adaptive radiation, but if you compare the Tasmanian wolf and the placental wolf, in their case, this is going to be parallel evolution. But no, you're making mistake here. Chali, next part. Next question, select the incorrect one. Vascular bundles of dicot stem said to be open. Why? Because they are having cambium in between. Intrafascicular cambium is primary lateral. Why primary lateral? Because when you look at the dicot stem, when you look at their vascular bundle, xylem is there, phloem is there. This is basically cambium. You can also call it as intrafascicular cambium. You can also call it as intrafascicular cambium. So all tissue lying inside the vascular cambium are called bark. So they are saying incorrect statement. out inside the vascular cambium. No, beta. And bark is the non-technical term. Endodermis is referred to as star sheath. So it is the C which is going to be the answer here. C is incorrect. Now next part, dash will show the adaptive radiation resulting from competition for food in a population. Resulting for competition for food in a population. Quick. Food in a population. Right, they were fighting for what? They were fighting for food. So it is going to be Darwin finches. It is going to be Darwin finches. Is that clear? So which of the following is not an example of XC2? Not an example of XC2. Wildlife sanctuary. Wildlife sanctuary is the example of in situ conservation. Wildlife sanctuary is an example of in situ conservation. Now, which of the following epithelium is most suitable for quick exchange? Beta, where there is the diffusion, you will go for simple squamous epithelium. That is going to be the answer. This is the important one. Just even if you talk about the blood vessels, just even the alveoli, simple squamous epithelium, blood vessel, endothelium is there. That is again simple squamous epithelium. Next question, next question, next question, next question. If a cell receives a signal to contract, then its neighboring cells are also stimulated to contract. This is due to the presence of, out of all the options, it is going to be intercalated discs. Where are you going to get that? In the cardiac muscles. That is why they are working together as a unit. That is why they are working together as a unit. Intercalated discs, like, you know, the gap junctions, right? They are fused here. The membranes are fused. So when the wave of contraction will pass, it will contract together like a unit. Okay. Chaliye, next question. Which is not a pair of poikilotherms? Poikilotherms, you understand? Poikilotherms mane you are talking about the cold blooded organisms here. They are saying not a pair, right? They are saying not a pair of. Yes, beta. देखो बच्चे, let's discuss that. Frog and your stingray, they are cold-blooded. Labio is cold-blooded, but Pteropus is your flying fox, right? So this is warm-blooded. So here also, this is not the pair which is of warm-blooded. And here also you will see Pristis or Carcadon is there. They are also cold-blooded, cold-blooded. Right, beta? Now, which is the first phase of growth? That is going to be, first phase is lag, then log, then stationary phase. Next, in which of the following the regulation of blood flow cannot be precisely regulated? Yes, everybody. Quick. Quick, everyone. Quick, everyone. Where is it not precisely regulated? Where is it not precisely regulated? The answer is simple, bacha. Even you can eliminate the odd one out. Even you can eliminate the odd one out. Ferritima, closed circulatory system. Labio, closed. Cuttlefish, closed circulatory system. Cuttlefish is a mollusca, but it belongs to that class of mollusca, right, beta? Cephalopods, yes, beta, cephalopoda, where you will see the cold-blooded, closed circular system. So mosquito, that is an arthropoda, open circulatory system. So that is why here the answer is mosquito. Next part, following are the role of predation except. Matlab, all are the role of predation except. All are the role of predation except. All are the role of predation except. Quick. Quick. Beta, it helps in maintaining species diversity. Yes. Is it a potent force of organic evolution? No. Predator act as conduits, money channels of energy transfer across the trophic level. True. Population of prey is kept under control by the predators. Absolutely correct, beta. The B is going to be correct answer. Next, which of the following is incorrect? Which of the following is incorrect? All animals do not depend directly on plants for their food needs. The death of the organism is the beginning of grazing food chain. Those animals that depends on primary carnivores for food are labeled as tertiary consumer. हाँ, ये primary producer in an ecosystems or phytoplankton algae. So this is direct. B is direct, that this is not the grazing food chain. Next beta, select the correct match for the following. Quick, select the correct match for the following. Everybody quick, select the correct match for the following. Select the correct match for the following. Estivation and example. देखो estivation, so here the correct answer is A, because yes, gulmohar is the example of imbricate. Gulmohar is जैसे aloe में you have valvate, so that is how you have to remember. Okay, so A is going to be our correct answer here, right? So here estivation is imbricate, example is gulmohar, that is correct. Next question, carbonic anhydrase enzyme, where do you find this? And it is the fastest enzyme. It is the fastest enzyme. So carbonic anhydrase, what's that, guys? It is the fastest enzyme. Fastest enzyme. Right, beta, we have already solved around 100 plus questions. Do you know that? We have already solved around 100 plus questions. Do you know that this PDF, if you follow this channel regularly, you know that I provided this PDF yesterday, right? And I asked you because they are all the NT authorized mock tests which I have picked up so that you can have more practice of PYQs, beta. Right? So more than, yes, more than 100 questions you have already practiced. I think it is 110 or plus, right? Because few slides only I added. Right, so yes, it is 1.5 hours, but look at this. You have solved many questions, and I know they are not that lengthy statements, but still. Okay, so carbonic anhydrase, they are mainly in RBC. Mainly in RBC. More concentration is in RBC. Not in plasma, beta. Not in plasma. Chaliye, select the incorrect matching. You know it very well. NACO. Right. Remember na AIDS Day is 1st December. AIDS Day is 1st December. Right. So NACO is the National AIDS Control Organization. ELISA is enzyme linked immunosorbent assay. SCID is the no beta. It is the severe combined immunodeficiency disorder, and PKU is phenylketonuria, which is autosomal recessive disorder. So here what's the correct answer that we have, guys? The correct answer that we have is C. Next part, which blood vessel end in an organ other than heart? Other than heart, obviously hepatic portal vein. Hepatic portal vein. Which site is the site for oxidative decarboxylation? Focus on the terms like whether they are asking decarboxylation or the phosphorylation. Or the phosphorylation. So they are saying which of the following is the site for oxidative decarboxylation? Decarboxylation is the word. Obviously, this is going to be the matrix of mitochondria. This is going to be the matrix of mitochondria. Right, beta? Right, beta? Chaliye, let's go to this. Pericardial fluid is a protective fluid found between. Pericardial fluid is a protective fluid found between two layers of. Pericardial is the word. Two layers of pericardium, of course. Two layers of pericardium, of course. Next question, a single turn of citric acid cycle will yield what? A single turn of citric acid cycle will yield. Let's solve like in next 10 minutes, we'll switch to the chemistry part, right? In next 10 minutes, we'll switch to the chemistry part. So you tell me, a single turn of citric acid cycle. हाँ जी बेटा, सिंगल टर्न ऑफ सिट्रिक एसिड साइकिल विल गिव अस 3 NADH2, 1 FADH2, 1 GTP. Do remember the steps. Do remember the steps. So mitral valve is present. You answer this. Please don't make mistake here. Please do not make mistake here. So the mitral valve is present between the mitral valve is present between the mitral valve is present between the beta mitral valve. You remember it is also known as bicuspid valve. Bicuspid valve. So it is in between left side. So this is important to remember. Left atria and left ventricle. Left atria and left ventricle. So B is going to be the correct answer. Next question. Identify the correct sequence of flow of electrons in the light reaction. Identify. Again, these questions are directly from the NCERT exemplars. Right, these questions are directly from the NCERT exemplars beta, right? So go for it. Go for it. हाँ जी, go for it. Beta, bicuspid valve. Bicuspid valve it is going to be. It is not valve. It is bicuspid valve. So identify the correct sequence of flow of electrons. So beta, in the case of Z scheme or Z scheme, PS2 to PS1 electrons will flow. So PS2, then you will have pheophytin, then it is going to pass that to the hydrogen or to the proton carriers. That is going to be your plastoquinone, which will make plastohydroquinone, which will pick up H positive even from the stroma. Then you are having cytochrome B6 F complex, and then you are having plastocyanin. PS1 will be there, ferrodoxin, unknown electron acceptor A, and then ferrodoxin. FNR is also associated with that. That is ferrodoxin NADP reductase. Then it will pass it to the NADPH. Is that clear? So what is the correct sequence? You will start with PS2, plastoquinone, cytochromes, PS1 or ferrodoxin. So D should be the correct answer. Again, NCERT exemplar. Okay, so if your plant physiology is not up to the mark, solve NCERT exemplar. There is a separate session for that, and the PYQs. Now, NaCl and urea is going to it is responsible for the gradient of medullary interstitium. Now, in the case of photosynthesis, light independent reaction, right? It is not light dependent. Light independent reaction will be there in the stroma beta. Why? Because it is the dark reaction that we are discussing. What is it? It is the dark reaction that we are discussing. So which is the not the function of angiotensin 2? Which one is not the function of angiotensin 2? Which one is not the function of angiotensin 2? Okay. Not the function of angiotensin 2. Obviously, beta, vasoconstrictor it is. Yes, ma'am. Increase GF, glomerular blood pressure. Yes, ma'am. Is it going to decrease GFR? No. If it will increase blood pressure, it will increase GFR. It is having vasoconstrictory effect. So it will not decrease GFR. So C is going to be our correct example. So cut vegetables and fruits. Right, beta. The cut flowers and vegetables can be kept fresh for a long period with the help of. Yes, everybody. With the help of. Quick, everyone. How can we keep it fresh? How can we keep it fresh? It is simple. It is going to be cytokinin. It is going to be cytokinin. Maturation phase of cell is characterized by. What is going to happen in that maturation phase? Speed up. Speed up, guys. Maturation phase is going to be characterized by. Maturation phase is going to be the characterized by. So in maturation phase, don't you think modification of protoplasm will be there? Of course. Don't you think thickening of cell walls will be there? Of course. So both A as well as B. Isn't it? Isn't it? So identify the incorrect pair. Identify the incorrect pair. Identify the incorrect pair. Go for it. Identify the incorrect pair. Speed up. Identify the incorrect pair. Puccinia, causative agent of rust, aspergillus, citric acid, astillego, rust fungi. No, beta. If one is rust, it is smut. Neurospora, extensively used in the genetics. Yes, bachai, that is true. So it is the C which is going to be correct answer. Done, beta. Now, goniolics belongs to, you know it very well, dinoflagellates. Now, which of the following center structure is basically for body temperature? Yes, answer this. That's again easy. That's again easy. Speed up, guys. Speed up, speed up. Yes, bacha. Which center? Temperature, thirst, and appetite. Exactly, exactly, exactly. It is going to be your high. Why are you not mentioning it correctly? It is going to be your hypothalamus. Body temperature is the word, right? So in an experiment in PBR 322, right, an alien piece of DNA is introduced using this cell one. The transformant will grow. The transformant will grow on. That's the word. The transformant will grow on. The transformant will grow on. So if you are using that particular enzyme, the transformant will grow on what? Right, beta, do you remember BTS? BTS. So tetracycline is having, this gene is having the restriction site for this BAMH1 and cell1. So you know that, right? This insertional inactivation of tetracycline will be there. So it is the ampicillin, right, on which it will be able to grow, beta. So answer this correctly. So this is all about the biology part, right? PDF is already provided to you. Few questions, right? Few questions from this part are left, right? We have solved, one second. Yeah, we have solved many questions. Tomorrow morning also we will meet for the quick revision of the PYQs, beta, right? So now you will go for chemistry. Okay, so solve chemistry. Okay, so bye-bye. See you all the best. Take care. Study properly. Now chemistry class will start. Done?
Good afternoon, good afternoon, good afternoon, people. Good afternoon, my NEET warriors. How are you? I hope everybody is doing fine. Please don't feel that level of anxiety. We are just less than 24 hours from the D-Day. So, again, don't be anxious, rather, practice with best of your efficiencies. So, good afternoon, Naren, Prerna, Ojas, Aryan, Yash, Marvel, Gamer, Sai, Karan, Ashish, everybody, Arhu, everybody. Chalo beta. Now, obviously, you have finished with you're done with your biology. So, without further ado, let us quickly go to the conceptual side, to the high probable side of chemistry, starting with organic chemistry. Acha, starting with atomic structure, a very probable question from atomic structure, how to calculate those angular and radial nodes. So the I will be setting the timer as 45 seconds. The first question is in front of you. 3, 2, 1, go, quick. List 1 is giving you the set of orbitals. And list 2 is giving you the number of radial nodes. Now, what is the formula of radial nodes? Sir, n minus l minus 1. What is the formula of angular nodes? Sir, L. Sir, it is a statistical formula, not a derived one. So please make sure. You remember these content properly. I hope everybody is good. Now, sir, what does this mean? Now you just have to calculate. Let's say if you just look at the option, A is either 4 or 3. And definitely because all options are saying 4. So what probably you should do, think of another option. So like let us check C. If you just check C is 3p. So radial node will be, so 3 minus 1 as in 1. What about angular nodes? So the value of L for P is 1. That means both are 1. So definitely radial node is also 1, and angular nodal plane is also 1. That means C is definitely 2. So C is 2, C is 2, C is 2, C is 2. That means you have your answer. Obviously, you have your formula. What about value of L? Sir, value of L is 0, 1, 2, 3. For what set of subshell? Sir, S, P, D, F. A very important point, beta. A very probable question. I hope most of you have got the correct answer for this one. Acha, percentage is less. So I hope now it is clear to people. Moving to the next one. Time starts now. Quickly, beta. Read through the question. The 50% of the question is done once you are reading that question properly. So please make sure you are reading the question very properly. Quick. Yes, sir. So 50% of the question is actually done. Yes. Hello, superstar. Now Neha, everybody is superstar here, because with the level of commitment which you people are showing, awesome. But I hope you are revising your content well and then practicing. Chalo, Hasini is saying acetone, writing that proper answer. So chalo, let's check beta. The question says I have C3H6. So that means definitely DU is 1 because CnH2n. So it can either be a cycloalkane or an alkene, sir, because it is giving an addition reaction, decolorizing the orange red color of bromine water. That means it has to be an alkene. So a 3 carbon alkene can only be propene with vicinal dibromide formation like this. So this will be my X. Now, after alcoholic KOH treated by a further strong base, which is sodamide, sir, this will make double elimination of HX and you will be getting your terminal alkyne, that is propine. And hydration of this propine, sir, first H plus will attack, after that OH will attack. So you will be forming, this guy will be getting H, this guy will be getting OH. So pi bond will be here, and here I will be having my CH3. Obviously, this is having one H, so this will be CH2. And then this guy will rather undergo tautomerization, where the cloud, pi cloud will be attracted here and hydrogen shall be tautomerized, and you will be getting your simplest ketone, which is nothing but acetone. That means answer is definitely option 1. Array, percentage is less beta. I hope everybody is clear now. Guys. Chalo, perfect. So obviously answer is acetone for this particular case. Moving to the next one. Again, probable one from atomic structure beta. Again, a probable one from atomic structure. Please read the question properly, guys. Read the question very properly, guys. Quick. Again, I am telling you, 50% of the question is done once you read the question properly. दीप्ति सिंह सर अब बहुत डर लग रहा है, क्या सोच के? हैं? क्या सोच के? Beta, don't feel anxious, rather work. Anxiety is bound to come, and sometimes, a bit of nervousness, a bit of anxiety is also good for for keeping the nerves active. So yes, that creates a level of goosebumps, and that is, once those goosebumps die in life, people start feeling monotonous. So rather, that's good. Bit of anxiety is always good. Bit, not anxiety, but yes, you should be having that level of newness in life, so that is okay. But yes, be productive with that, beta. So the which transition in hydrogen spectrum will have the same wavelength as the transition 4 to 2 of helium spectrum? Sir, definitely we should know this, that the wavelength will be same, therefore 1 by lambda would have to be equated, which is nothing but r into z square into 1 upon n1 square minus 1 upon n2 square. Sir, if I want two wavelengths to be same, then obviously R would be getting cancelled. Z for helium will be 2, so this is 2 squared. The transition which is talking of is from 4 to 2. So either 4 to 2, 2 to 4, sir, wavelength will be same. If I absorb that packet, I will jump from lower energy to higher energy. If I throw or emit that packet, I will jump back from higher energy state to lower. But yes, sir, the magnitude of wavelength has to be positive. So we rather take n1 as the smaller one. So this n2 is the smaller one. So 2 squared minus 4 squared will be equal to, we have to check for hydrogen. So for hydrogen, Z is 1. So this is 1 square, 1 upon N1 square minus 1 upon N2 square. These N1 and N2 are now transition states for hydrogen atom. So definitely do the math, because you just have one equation. So what I can do is this 2 squared will be multiplied. So this will become 1 upon 1 squared minus. So 4 squared is 16, 2 squared is 4. That means this is 1 by 4, and 1 by 4 can be written as 2 squared. So this I have on the left hand side. This I have on the right hand side. So you just tell me what can be your N1 and N2, sir. Obviously from 1 to 2. That means 4 becomes your right answer. 66%, awesome. An NCERT beautiful question. I hope everybody is with me. Moving to the next one. Quick. You have to match the column. Here you have chlorobenzene, they're forming phenol. So technically, because we know, because of the partial double bond character, no reactions of aryl halide are same as alkyl halides because of this partial double bond character. But if even if we want a nucleophile to attack this positivity, which is rather very difficult, then what I can do is I can supercharge that nucleophile. How to supercharge that nucleophile? Beta, by giving it more energy, more temperature, high pressure. That pressure and temperature will definitely create, in this case, phenol. But because you are providing OH minus, that means that medium will be basic, and because medium will be basic, so what will be formed? A sodium phenoxide. So that after acidification will give you phenol. I hope everybody should know this much of logic. And this process is nothing but Dow's process. So I hope you know what is Dow's process now. So that Dow's process is mentioned at fourth position. So A is definitely fourth with the proper temperature and pressure. So you should definitely remember this. That means A is fourth. That means A and B is gone. Now B you do not have to check, and if you just check for D, which is formation of picric acid, then sir, this, because this benzene ring is substituted with electron withdrawing group at ortho and para position, so rather this positivity is significantly increased, and that's why now even a weak nucleophile like water will be having interest. That means you just have to heat with water. That means D is definitely 1. That means 4 is your right answer. I guess this beautiful NCERT fact is known to everybody now. and everybody has revised that, but people are saying 50% C. So I hope the problem is resolved and now everybody is with me on the conclusion and on the conceptual understanding. So is everybody with me? Quick. Okay. Acha, people are answering. People are answering D here, but in the poll I have observed 50% were saying C. So I hope everybody is clear now, guys. Chalo, great. Yes, sir. So Arya Singer is saying yes. Thank you. Iron Man is saying yes. Go, Iron Man, go. Great. Sir, how to calculate energy from the equation of hydrogen? Beta, we were not actually trying to calculate because one equation can't give you two unknowns. Yes. You can do the observational math. Sir, what N1, N2 you have to pick as per options, as per the data. Sir, that's why I've done 1 squared and 2 squared. So yes, you can have proportionality. It can be any energy level which shall fall under that ratio also. So that is actually when once we discuss this in as a topic, it is law of equipartition of energy level. So practically whatever transition will be happening from N1 to N2 in hydrogen, it has to be multiplied by Z of whatever species you are talking of. So let's say 2 to 4 of hydrogen will be what transition? For helium, sir, helium Z is 2. So 2 into 2 is 4, 4 into 2 is 8. So in helium 4 to 8 transition will give you the same thing. Why? Just simple observational maths beta. है ना? Chalo. I hope everybody has got answer to this one as well. Yes. So percentage is great. 60%. At least the percentage has increased. Now let's check why is it so. Identify the ion hydrated in solution which is not, beta, please make sure you're reading such stuff, not correctly matched with its spin only magnetic moment in Bohr magneton. So let's say we have to check. So let's start checking. So chromium is 4s1 3d5. Yes, you can do such things in your mind also, but because I am trying to explain also, because the percentage I saw is 40% could not have attempted it properly, so that's why. So that means that this is 3d3. So sir, whatever be the ligand, if it is 3d3, then this 3d3 is bound to have the three unpaired electrons, sir, and those three unpaired electrons will be creating what magnetic moment, sir? Sir, under root of n into n plus 2 Bohr magneton, sir. So value of n is 3. So 3 plus 2 is 5. 5, 3 is 15, sir. So that means it is less than 16. So less, approximately around 4, less than 4. So it is saying more than 4, sir. Obviously, this is incorrect, sir. So that definitely this is my answer. Yes, I am telling you, in your examination, such things may never happen. I mean, may or may not happen that you are following that case in the first option only. But yes, rather sometimes if it is in D, then you have to check through all the options. So that's why keep a clear eye on the options that are given so that you can manage your time efficiently, because this is bound to happen for your inorganic questions where you have to check through all the options. I hope everybody is clear. Yeah. Moving to the next one. Time starts in 3, 2, 1. Go. Quick. Quick, beta, quick. Quick. Shahnawaz Qureshi saying, sir, take it fast. Beta, have compassion. What about the remaining number of 40% students who have incorrectly answered it? And maybe, beta, maybe even in your exam, you could also, miss some points. So that's the reason for having an active discussion. So yes, some level of explanation has to be there, because that's why I wait to at least see the poll, and then only I decide how to, to what level we should approach the problem. I hope everybody is clear. Let me just put this. Chalo, I hope everybody is clear. Great. A, people are answering. A, let us check. A. Ah, great. Yes. So A is definitely right answer. I hope you must have read the statement properly. The question is saying which is not correct. And then it is asking what is correct answer. So what does this mean? That among them, what are the statements that are incorrect, and whoever is incorrect, now you have to give the correct answer, that whoever is incorrect. So make sure you are reading the statements very properly. Now, sir, obviously that means you have to mark the incorrect statement. A is saying for water, magnitude of Kb is more than magnitude of Kf. Sir, definitely incorrect, because Kf is 1.86 Kelvin kg per mole, while value of Kb is 0.52. So, sir, obviously this is incorrect. So, because this is incorrect, hence an answer. So, I need A in my, obviously all the options are having A. Now, the elevation in boiling point of water when a non-volatile solute is added to its larger in magnitude than its depression in freezing point. Sir, come on yaar. Delta, if concentration is same, delta TB will be equal to KB into concentration. Sir, concentration you do not have to check, so definitely the magnitude thing will be going on KB value and KF value. Because KF is more, so beta depression in freezing point will be more. It is saying elevation in boiling point is larger. So definitely this is also incorrect. So B is also my answer. So B is also my answer here. The A and C are probable. Now I just have to check Delhi or C. Or Delhi, because C is not even there. So Delhi is saying the dimerized form of benzoic acid is. Sir, this is intermolecular hydrogen bonding. This is also intermolecular hydrogen bonding. So definitely we knew, sir, carboxylic acid, they tend to dimerize. So this is also correct. So correct, so not an answer. So D cannot be taken. That means A is your answer. I hope now everybody is clear. Guys, chalo. Moving to the next one. Yes. So time starts in 3, 2, 1. Again, beta, a highly probable logic from the chapter of solution and colligative properties. A high probable logic, quick, from solution and colligative properties. You are given a 0.1 molar weak acid and its pH is 2. So if its pH value is mentioned, you have to calculate the osmotic pressure. Sir, osmotic pressure pi is equal to Van 't Hoff correction factor ICRT. Yes, perfect. So people are answering beautifully in the comment section. Great. So let me just allow every student to attempt, which most of you have, I guess, attempted correctly only. So time is about to end. Let me just see. 64%. Aisha, Rutu, Badal, Sridhar. Beta, you guys are taking 6 seconds. Come on. Oh, great. I hope you are doing it with all due diligence and all due level of integrity, because that is how it is only going to help you. Chalo. So, sir, concentration C is given, sir. It is monobasic acid, as in it is type of HA, sir. So, it will be giving H plus, sir, and A minus, sir. So, if this is C, then C minus C alpha, this will be C alpha, this will be C alpha, and pH is 2. So, pH is 2. That means H plus ion concentration will be 10 raised to the power minus 2, because pH is minus of log of H plus ion concentration. So that means this H plus ion concentration, whose variable form will be C alpha, is actually given to us as 10 raised to the power minus 2. That means, sir, because this C is also known, which is 0.1 as in 10 raised to the power minus 1, that means, sir, I know alpha, which is 10 raised to the power minus 1. Sir, that means I can calculate correction factor, which is 1 plus n minus 1 times of alpha for a case of dissociating species, which is this weak acid. So value of n is, one molecule is breaking into two, so n is 2. That means this is 1 plus alpha. Alpha is 0.1. That means I is 1.1. Now we just have to put this in the value in the formula of osmotic pressure, which is ICRT. So I value is in front of us, sir. It is 1.1. Concentration also is in front of us, sir. It is 0.1 times of RT. That means this is nothing but Bombay. A practically and logically great one. But I hope now everybody is good. Guys, सर पोल नहीं आ रहा नेहा चौहान बेटा काइंडली जस्ट update your pylins app, because with that you can definitely participate in poll also and it will otherwise be helping a lot. आज की तरह अपना farewell. Okay, so Deepthi, welcome. And yes, I hope you will be loving this farewell, because after today, from after tomorrow, you will be that great doctor for which you are aspiring to be, for which you are putting all your efforts. So all the very best, beta. God bless. But yes, till that time, keep your nerves calm, keep your head cool, and give your everything in the moment, because whatever is present is inevitable. Whatever will happen will be decided what you are doing right now. So please, the class duration beta, just I guess I'll be finishing around 4, and then you'll be having a physics, and then obviously you will be there with you. Moving to the next one. Chalo beta, again a beautifully probable question, because a very famous organic compound, sir. What is that very famous organic compound? Mesitile oxide. What is this mesitile oxide, sir? Sir, it is having 4 carbon chain and it is having a ketonic group. Sorry, 5 carbon chain. That means, sir, I need pent. Sir, I need ketonic group at the second carbon. I need an unsaturation, sir, between the third and fourth carbon, and at fourth carbon I need a methyl. Sir, how to remember this? Beta, this mesitile oxide is actually a aldol, a self aldol product of acetone. So do you remember acetone? Yes, sir. What is acetone? Beta, acetone is CH3COCH3. So do aldol of this, sir. How to do aldol? Beta, this carbon will be turned into a nucleophile CH2 minus. It will be attacking its own species, and the another carbonyl carbon will become OH. After that it will lose OH. This will again lose H. That means, sir, its 2 hydrogen is gone, and a double bond will be created between this and the carbonyl group of the the other molecule, which is carbon. So this is, let's say, carbonyl carbon. This is CH3 and this is CH3. So this is how you should remember. This is how you should remember. Acha, haan. In that case, beta, I guess I have just made this incorrectly. This should be 3, 4, na? So that means you should have your pi bond over here. So how to name this, sir? Because functional group is here. So 1, 2, 3, 4, and 5. So at fourth position you have methyl. At second position you have ketone, and 3, as in between 3, 4, you should have your unsaturation. So this is nothing but your mesitile oxide. You just have to check. So I guess this is given in option 1 also, option 2 also. Okay, only option 1 and 2. Then you have to give IUPAC of oxalic acid, sir, 2 carbon dicarboxylic acid, as in C double bond O, then OH. Sir, C double bond O and then OH. Sir, this is my ethane 1, 2 dioic acid. We do not generally take 1, 2 because these carboxylic acids are always terminal functional group. That means option A is nothing but your right answer. 83%. Great. Awesome. Awesome and blossom. Moving to the next one. I hope everybody is doing great. Everybody is revising their content, because beta, these are the probable logics. I'm not saying the questions will come. Let us be practical with it. But yes, sir, among them only they will come. This will come. Among them it will come. Yes. Why? Because, sir, they can't ask anything beyond NCERT. Cool. Cool, cool, cool. Okay. G, sir. Yes, sir. Are you saying sir? Yes, beta. Yes, are you? B. Just eating apple. Chalo, great. B, B, B. B, sir. B. 72% are saying B. Great. Sir, the total number of donor atoms, sir, that means you just have to calculate coordination number, sir. So who is my metal? Sir, platinum. In what oxidation state? +4. How many ligands? Bis as in 2. But obviously a polydentate ligand. And what is this? This is ethylene diamine. That means this is NH2, CH2, CH2, NH2. So one ethylene diamine will be ligating via 2 donor atoms. So 2 will be ligating by 4. So this will be giving 4 coordination number. But I have 2 thiocyanato as an SCN minus. So more 2, that means sir +2, that means sir coordination number is 6, sir. Yes, this is, and thiocyanato is a ambidentate ligand, but sir at one point only one will ligate, sir. Yes, it can ligate through so it can have linkage isomerism, but obviously over here this is just NCERT fact, which is coordination number 6. I hope everybody is good. Moving to the next one. Time starts now. Quick. 1 mole of ethanol was completely burnt in oxygen to form CO2 and water. You have to calculate enthalpy change of the reaction. Enthalpy of formation of ethanol, CO2 and water is given. Values are there. Beta, please make sure you are calculating properly. Sir, shall we do such questions in the first go? Never. Sir, shall we do these questions in the second go? Yes. Sir, what we should do in the first go? Beta, all the inorganic, organic questions which you can, even physical questions which you can attempt without writing within 3 lines. You just have to write maximum 3 lines. If, sir, taking more time, leave. Sir, if we keep on leaving such questions, don't worry. First, see through all your 180 questions. You will be also given those extra 15 minutes this time. I'm not saying they are very much, but yes, beta, they are more than 3 hours. They are more than 180 minutes. So you will not be getting 180 minutes. You will be getting 195 minutes. So 195 is more than 180, mathematically correct. But sir, I have to do this, that. Everybody has to do this, that. So whatever you have, make best utilization of that, because that is inevitable. Great. Sir, will you eat mango? Yes, Saloni Singh, I will eat mango. Although mango, I mean, mango is a fruit which is just full of loaded with calories, huh? Chalo. But who is saying calorie? Master, you are already mota. So that's fine. Huh? I do not want to become much mota. Chalo, great. Go, D. Chalo. Go, D. Arey yaar. Percentage is very less, beta. Maximum of the students are saying Bombay. Chalo, let's do this. Sir, you just have to write the reaction, sir. Combustion, sir, means C2H5OH, which is ethanol, sir. It is combusting, as in reacting with oxygen, and then it is bound to form CO2 and water, sir. Sir, what you have to do is just balance the reaction, beta. Balancing is a class 10th job, is a class 9th job. Yes, if you do that properly, your answer is in front of you. Then do the stoichiometric maths. Sir, 2 carbons, so I'll be having 2. Sir, 6 hydrogens, so I'll be having 3. Don't bother, but still 4 plus 3 is 7, so this is 7. No. 1 is here, so this leaves 6. So I need 3 over here, because 3 into 2 will be 6. So this is my balanced equation. And enthalpy of reaction will be whatever standard value will be, summation of enthalpy of formation of products minus summation of enthalpy change of formation of reactants, because we are doing at standard conditions. So standard now, that means for product I will be having 2 into of CO2. The value of CO2 is minus 393. So minus 393 plus 3 times of water enthalpy. So water enthalpy is given as minus 286. So minus 286. This is product, sir. Minus what about reactant, sir? Ethanol, sir. 1 times of ethanol. So minus 277. And sir, if you have element in its pure or native state, oxygen is present as O2, so its enthalpy of formation is 0. That's why not given. Now after this is just mathematics, beta. No one can help you, but your practice can help you to do such questions. I'm not saying this is easy to do in an examination, but yes, it is easy to practice right now. So the more you practice right now, the better your chances will be to attempt and do those calculations in the D-Day also during the examination. So I hope everybody is now clear with the logic. Moving to NCERT concept and NCERT understanding of electrochemistry, that too from the side of batteries, sir. Electrochemical cells, you have to learn what are the cathodes, what are the anodes, and what is the electrolyte and what is its practical use. This question corresponds to all those factual features. If you just know, with the fuel cell that we have, it was initially used in the space mission by ISRO, the Apollo, and that's why, sir, the hydrogen cell, which is using hydrogen and oxygen as a fuel, creating water as a byproduct, that is also used for other purposes by the astronauts. So definitely D goes with 2. That will definitely give you an answer. D goes with 2 definitely gives you an answer, but I'm still saying, beta, please remember, for all these elements, for all these batteries, you should know, sir, their cathode, their anode, their practical uses, whatever is that NCERT fact. है ना? So moving to the answers, 82%. Great, great, awesome. Chalo. Moving to the next one. Time I have started. Now everybody, please try and answer this one real quick. Quick. Sir, I am scared of apple. Don't eat then. If you are scared, then don't eat. Acha, but beta, an apple a day keeps a doctor away. Okay, that's why you're scared of apple. Great, yaar. An apple a day keeps a doctor away. If people start keeping doctors away, then where you will be going? Eh? Say in that case, equation 1 is an apple a day keeps the doctor away, है ना? And equation 2 is uh, if you are well, you will never need a doctor. So equation 1 and 2 says doctors should hate apple. So any justification, future doctors? Any such a polite and great teacher. Thank you, Saloni. Ram is saying, sir, I dash. I will not read that, Ram. Bad, beta. Don't feel. I mean, you should, beta, respect your hard work. Chalo. Apple should be banned. Okay. Because an apple a day keeps a doctor away. Chalo. Now, I hope people has answered. Yes, good. Beta, a very probable NCERT fact. Sir, this is an ester. So excess of Grignard reagent first will give the nucleophile CH3 minus. It will go. Sir, pi bond will move. Sir, lone pair will again go and will kick out the alkoxide group. So first ethyl added, sir. But sir, it is still a ketone. So the second one will also go and will convert after acidification, will convert it into a 3-degree alcohol. So definitely here I'm going to have my 3-degree alcohol. Now, what about this, sir? Same thing, sir. This is also an ester, sir. Here phenyl will go, sir. Then, sir, the pi cloud will move. Then the lone pair will again go and will kick this alkoxide ion. And then, sir, but still it will be a carbonyl group. So second molecule will go, and after acidification, it will be alcohol. But sir, after this is gone, this carbon will be just having two alkyl groups, as in two aryl groups. So it will be definitely happening 2 times. But because this is an aldehyde, not a ketone rather, so your alcohol will be 2-degree. Sir, what about here? So here also, because I have hydrogen, although this is an ester, so this will also be happening twice. But still, after happening twice, you will be forming your 2-degree alcohol. Yes, sir. This is a carboxylic acid. So first this OET will be kicked, and here also 2 times, because after carbonyl, the another methyl will be going in. So here you will be having a 3-degree alcohol. So definitely first and fourth, as in Delhi, will give you a right answer. Logically sound one, beta. Moving to the next one. I hope that was a beautiful NCERT question. That was a beautiful NCERT concept, I guess. Not question, but yes, a very probable concept. Chalo. Great. Great. Would you repeat? Okay, somebody was saying, if sir, repeat. Repeat previous one, beta. Hai, beta? Sir, can we expect the same model questions? Yes, beta, you can definitely expect that. That's why we are doing this again and again. Sir, you sound like, hey, I want to read that, yaar. Hey, yaar, I want to read that. Hey, I want to read that. Acha. Thank you. Hey, you know what? I love reading these comments. Sir, अरे यार, Ashish is after apples. Yes, Saru Latha. You can expect these, beta. Definitely you can. Chalo. I guess, acha, let us do it. The correct boiling point of the compounds given below. So, sir, for boiling point, you need intermolecular forces of attraction. What do you feel is the strongest intermolecular force of attraction? Beta, strongest intermolecular force of attraction is H bond. Sir, it is hydrogen bond. Now, what is that hydrogen bond? Where you feel that hydrogen bond exists? Sir, it definitely exists in alcohol, because this is a alcohol. So this alcohol will definitely, this guy will definitely associate with hydrogen bonding. Although all their molecular masses are comparable, and once masses are comparable, you only think of intermolecular attractions. Strongest is hydrogen bonding. That means, sir, B shall be the having the highest boiling point. Now, among aldehyde and ketone, sir, because ketone will be having more directed polarization. This a complete thing is giving electron, and that guy is taking electron. So the minus and plus segregation is more organized here. So it will have a better dipole attraction, and because better dipole attraction, so better extent of intermolecular attraction. So that's why boiling point will be slightly more for this guy. So that means D will be more than C, and obviously this is a weak dipole attraction, although it is not non-polar. But yes, these ethers are having weak dipole attraction, because OC has got that delta negativity and delta positivity, but rather quite less than the carbonyl group. That's why it will be weakest dipole attraction. So your answer is definitely Bombay. 49% of you have answered it correctly, so great. I hope now everybody is clear. Moving to the next one. Time has started. The classic formula, the classic concept based question from what chapter, sir? Beta, from solution, from mole concept, the basic interrelationship between concentration terms. If you are Chandan, then I'm water. Okay. Great, beta. Association factor. Lilia is asking, can you tell me, beta, for solution chapter, for in case of dissociation, this correction factor is 1 plus 1 upon n minus 1 times of beta. This n is for if a species getting dimerized like carboxylic acid, so directly put this n as 2. So 1 plus 1 upon n minus 1 times of beta, where n is for dimerization 2, trimerization 3, tetramerization 4, although you only say case of dimerization in your NEET examination, and beta is the degree of association, like the degree of dissociation. So that this is your formula for association in case of association in case of Van't Hoff correction factor in the chapter of solution. Now, so here the formula used is density is equal to, sir, molarity into 1 upon molality plus molar mass of solute upon 1000. I hope everybody remember this formula. So density is given as 1.12. This has to be in gram per ml or gram per cc, so kindly validate the unit. Then density of x molar, so molarity is x, sir. 1 upon molality is given, sir, 3 plus who is my solute? NaOH, so 23 plus 16 plus 1 is 40 upon 1000. Now rest is just mathematics. If you just solve, you will see, because that's a dilute solution, so molality and molarity will be same. So Bombay is going to be your answer. 51% of the students have cracked this one. So great. Moving to the next one. Time starts now. Quick. Sir, if you are pearl, I'm your thread. Okay, gaming. If you're doctor, I'm your teacher. Pearl and gaming. Gaming bow. अल्डिहाइड का काम कहाँ है? और केमिस्ट्री में बेटा. चलो. Sir, I'm in 7th rank. Perfect, Ashish. Great. Sir, generally ether has more boiling point than hydrocarbon. Yes, ether has more because ether is polar, na? Hydrocarbons are non-polar. So if you compare ether with hydrocarbon, they have, so in comparison to hydrocarbon, hydrocarbons are attracted by weak London dispersion forces. They are rather attracted by strong dipole-dipole attraction. So yes, if you compare ether with hydrocarbon, hydrocarbons generally have higher boiling point. Chalo. So I hope people have done and attempted this as well. Let me see the percentage. 60%. Chalo. Great. Now, अरे यार, very good यार. Come on. Now, the question says, for a reaction, the graph of ln k on y-axis and 1 upon t on x-axis is a straight line. So, for your chemical kinetics, you have to give that activation energy. Sir, we know rate of reaction is equal to the constant times of minus E upon RT, but because we are interested in ln, so ln k will be equal to, sir, ln of A minus EA upon R into 1 by T. Now it is saying this you should consider as x, this you should consider as y. So that is, sir, my value of slope, which is mx, is nothing but this. So my slope is minus EA upon R, which is, I guess, given to me as minus 2 into 10 raised to the power 24. So minus minus gone. If I'm taking R as 8.3, so my activation energy will be 8.3 into 2 into 10 raised to the power 4. And you have to give this activation energy, but in kilojoule. So I have to divide this by 1000. That 1000 will kill these 4 power, and this is 83 into 2, so 8 2 is 60, so 160, 160 as in 166 becomes your right answer. So a probable one, beta, a probable one from chemical kinetics, a high probable logic from your chemical kinetics. I hope everybody is clear. I hope everybody is clear. Good. Great. Great. Quick attempt the next one, guys. Attempt this next one, guys. Ether is less than hydrocarbon. नहीं beta, in for see beta, for hydrocarbons and ether, because we know some alternation effect also in hydrocarbons, that hydrocarbons, as the carbon chain increases, for the melting point, I'm saying, the even number of carbon bearing alkanes are more symmetric, therefore they generally have high melting point. Yes, if you just compare I'm not speaking specifically for two. There may be a hydrocarbon whose boiling point may exceed that of ether. Why? Because as carbon chain increase, boiling point will also increase. So generally we do this for species which are having comparable mass. And whenever species are having comparable mass, then what priority we give? We give priority to the intermolecular forces of attraction. But yes, if at all there is some specific case, as we are speaking as per NCERT, but if you generalize, ethers are slightly polar, therefore these ethers, these alkyl halides, they definitely have more tendency of having more boiling point. Why? Because they are slightly more polar. Why I'm saying slightly? Because these carbonyls are highly polar, so their dipole-dipole attraction is super strong. है. So I guess I have answered a student who's the same question he's asking 2-3 times. है. Chalo, perfect. Now, I hope everybody has answered to this one too. 72%, great. The question says, pair of elements with the same number of electrons in their respective 4f. That means that they are talking of lanthanoids. So atomic number is also given, but you should know that rhyme also. So let us first check europium, or rather all are written, so let us remember that rhyme. Sir, after lanthanum is, sir, 57. So after 58 we will be starting with lanthanum is 56, 54, 55, 56, 57. Yes. So after 58 you will be having, after lanthanum, सीना पार नदियां पीएम की समाई, यूं गदगद तब दिल ये हुआ, अरे तुम ये भी ल्यूटीटियम लूट लो. So that means 57 plus 14 is 71, sir. So it will be ending at 71 and this will be 58. So that means, sir, this is sir 6s2 5d1, and this guy will be 4f1, and that guy will be 4f14 with some basic irregularities. Now, sir, after this, what we have to do, sir? Now 72 will come next to lanthanum in the D. So 72 will be here, sir, and you have 72 also given in the question. That means you have hafnium over here. I hope everybody is clear till this. Now, you are supposed to give those elements which have same number of electrons in F. So let us compare, sir. Then who is A? A is europium. What is europium, sir? 63. That means after 58, how many electrons you have to add, sir? 5. So add 5, sir. So if I add 5, this will be 4f6. and 4f6 can call one electron to make fully half-filled configuration. So this European EU will be having 4f7. I hope everybody is clear now. What about GD? GD is just, sir, one more. So if I just need one more, why I will call this 5D1? This will be having 5D0. But why I have called this 5D0? Because I wanted my inner shell to be half filled. Once the next element, which is GD, is already having that extra electron, so this will be 5D1 and 4f7. But sir, 7 is having, the 4f is having same 7 electrons, so that means, sir, A is definitely my answer. Now, similarly, after this, sir, what is YB? YB is 70. So I'm intentionally going there because that they will be having more probability of having all the fully filled F subshell. So, sir, this is 4F15, which is LU, which is 71. So what about 70, sir? Beta 70 will be YB, ye terbium. And that means that this guy can also call, so this will be having 5D1. This will call the 5D electron, so this will be 5D0 and 4F14. So these two also have same 4F. So, acha, these two definitely have, but here YB and HF is given. So where is my HF? HF is here, hafnium. Hafnium will be, sir, here 5D2 will be filled. So this will be having 6s2, 4f14 and 5d2, sir. So 5d is 2, sir, but 4f is still 14, sir. So obviously all these after hafnium and all these elements will have same electrons in 4f subshell, which is 4f is completely filled. So definitely they too also, that means, sir, A and C, and only one option is giving you that A and C. And then yes, you have to check for all these options, but beta, in organic chemistry, the more you practice, the more you remember things over the head. And if you remember, you can directly recall these things and it will take less time. But yes, if you do not recall this, if you do not know this exactly, if you do not remember this, then you have to give a bit of time to such questions. So rather attempt these inorganic questions also in the end, not at the start. है ना? Chalo. So I hope everybody is clear. Moving to the next one. Time has started. Quick. The ratio of sp3 carbons to sp2 carbons in the product P of the given sequence. So quick. Ratio of sp2 and sp3. Quick. Quick, quick, quick. B, B, B. ओके सर, आपकी वॉइस बहुत प्यारी है सबरीना, थैंक यू बेटा. It is you who's rather more great beta, because it is these teachers, it is these students which make us what we are, else we are nothing actually. Chalo, great. Attempt this one beta. 3:1. Shatrughan. Shatrughanna. Mr. Maji is saying D. Great. लीशा सर, क्या पढ़ा रहे हैं? टाइम पास बेटा. Come on यार. Biology, physics, chemistry, RE-NEET 2026. Acha, okay. Then if you are serious beta, then I'm answering. We are taking the high probable questions. High probable questions. Chalo. Chalo, I hope time is also done. Yes. अरे यार, percentage is less यार. So Neha, Pranshu, Raj, Kanwan, Satish, Abhishri, Mariam, Roshan. Great, yaar. Great, great. Awesome. Chalo. Now, let's explore, sir, the ratio. So first, let us form the carbon and then we'll see the sp3 and sp2. Sir, this is aryl halide, sir. Sir, this is acid chloride, sir. Anhydrous Al3, sir. This is Friedel Craft alkyl acylation, sir. So this acylation will be happening where, sir, although this is electron withdrawing, sir, but the directive influence electrophile is definitely produced because you have an Lewis acid. So at para position, you will be having your major product formation. So this is para substitute chloro at para position, chloro substituted acetophenone. Now with Clemenson reduction, this guy will be reduced. What guy is at the carbonyl group? This carbon will be turning into an alkane. So how many sp3 hybridized carbon you have? Sir, this is 1, this is 2. So technically, sir, I have 2 sp3 hybridized carbon. So sp3 hybridized carbon are 2. And these sp2 hybridized carbons are 6, and you are supposed to give the ratio. So that will be sp3 to sp2. So this will be 2:6, as in 1:3. So Delhi is your right answer. I hope everybody is clear. Moving to the next one. Yes, a good one, a logical one, from a probable one rather. Probable one from chemical bonding. So the structures predictability. So anyway or anyhow, they are definitely going to ask you regarding the structures. And what are these structures, sir? They can only be predicted as per valence shell electron pair repulsion theory. So if you just apply your VSEPR theory, I guess XeO3, sir, each oxygen will require 2 electrons. So 6 utilized, sir. So 1 lone pair, sir. So 3 bond pair, 1 lone pair. This will be trigonal pyramidal. This Xe having 1 lone pair, sir. 1 double bond O, 1 double bond O, 1 double bond O. So this is trigonal pyramidal. Now, who is having that trigonal pyramidal geometry, sir? Obviously, ammonia. So that means A is 2. So only this option is gone. What about XCF2, sir? 2 bond pair utilized, sir. So lone pair will be 3. So 3 bond pair and 3 lone pair and 2 bond pair. So steric number 5, sir. Lone pair will be at equatorial position, sir. So Xe will be linear with the lone pairs occupying the equatorial position, and axial fluorines will be there. So technically it is just linear, sir. So who among them is linear? Obviously, sir, I3 minus. This iodine having 7 electrons will gain 1 more. So it is now having 8. If it is forming 2 bonds, sir, so again, sir, 2 bond form. So out of 8, 2 gone, sir. That means again, sir, 3 lone pairs are 1, 2 and 3. That means that this is same as the case of XeF2. That means B is definitely 3. So B is 3. B is 3. That means this is gone. Now we just have to check C. So this is oxygen this will take 4. This will take 2. So 4 and 2 is 6. That means it is just having 1 lone pair. So this is 4 bond pair and 1 lone pair. That means it will be a seesaw shape. So xenon having 1 lone pair, sir, and 4 bond pairs, sir. So double bonded oxygen, so fluorine and fluorine. I hope everybody is clear. I hope everybody is clear. That means that this is nothing but seesaw shape. So where I have 4 bond pair and 1 lone pair are in SF4. So definitely this C goes with 4. That means C is your answer. That means C is your answer. A very probable question, beta. 80% of you are correct. But yes, please make sure you are confident. You are very, very confident with your conceptual understanding of VSEPR theory so that you can attempt these questions at faster speed. Quick. Rather, in order to check the percentage ionic character, sir, for an ionic compound, how do we check it? Sir, you calculate the percentage ionic character by the dipole moment that it is actually showing in nature. upon the dipole moment that you calculate from your formula. Sir, what is the calculated value of dipole moment? Beta, calculated value of mu is charge, the magnitude I'm talking of. Sir, it is charge and distance between charge. Sir, distance, sir, if I'm a molecule AB, then this is, let's say, delta positive, this is delta negative. So bond length will be the distance between charge, which is given as 1.41 angstrom. So this is 1.41 angstrom as in 10 raised to power minus 10 meters. But I guess here it is in SI, CGS unit. So in centimeter I have to convert. So this will be minus 8 centimeters. So in CGS system, as in centimeter, I have taken the bond length. Now, sir, what is the charge, sir? This is partial plus partial minus. So plus 1 plus minus. And this is one magnitude of one charge, as in for one electron, this is 1.6 into 10 to the power minus 19 coulomb. But sir, because it is the CGS unit, so this will be 4.8 into 10 raised to the power minus 10. If you just multiply it, I guess you will be getting your mu, and that mu value will be mu calculated. Let's say whatever ESU centimeter, let's say you are having some value me calculated. Yeah, into divide you have to do beta. Now, the dipole moment actually given is this. So this will be something, some A into 10 raised to the power minus 18. Now, how to calculate this value? Sir, actual is given 0.78. So calculated value will be A. Just transform this as 10 to the power minus 18. It will be cancelled and then multiply by 100. If you just do that, I guess most of you have already done it. I guess this you will be getting somewhere around 11.5. So where is it? Here. I'm not saying the calculation is easy, but yes, beta, please do remember a high probable logic from either you can say chemical bonding, either you say from GOC, but yes, a very probable logic that you should know. I hope everybody's clear. Chalo, perfect. So let me just check. Great, 78%. Great, beta. I mean, people have calculated also. Great. Moving to the next one, and the time has started. Quick. Observe the following molecules. Sir, NH4 plus, you have to give number of bases. So in what is a Lewis base, sir? Sir, Lewis base will give electrons, sir. If I want to give electron, I should have those lone pairs. Sir, this is electron precise, sir. Complete octet, sir. No availability, sir. Yes. This has lone pairs, sir. So this will give, sir. So this will be Lewis acid having deficiency, sir. This can definitely give, sir. So this is the second thing. So this is also electron deficient. This is also electron deficient. This, sir. So carbonyl can also do, sir. Why? Because oxygen will burn once coordinate bond is formed. This carbon, after formation of coordinate bond, it can be thought of a covalent bond only. So donor atom will get plus, this acceptor atom will get minus. That means, sir, this carbonyl, this carbon is a very good ligand, sir. It is very good base, sir. So this is also good. Then here this can donate pi cloud. This can definitely donate pi cloud, but now only toward an electrophile. Ultimately, this is also because of this sp hybridized carbon. This is also it, but yes, rather it can act as a pi donor ligand, but not as a, you cannot say as a base. So definitely not taken. So if this is not taken, then how many you have? 3. So that means Bombay is your right answer. 41%. So I hope few of you have taken C2H4 also. Why not C2H4? Because carbon beta is sp2 hybridized carbon is electronegative, so it is not having tendency to give. Yes, if at all an electrophile is formed, then yes, towards that electrophile the pi cloud is attracted. If you're having a strong metal, then to that metal I can give my pi cloud as a ligand, but because of the high polarization power of the metal, I am not willing to give. But yes, if somebody is attracting me to such an extent, then I can think of giving. Like it does think of giving during electrophilic addition and during formation of that ferrocy, that pi complexes, pi ligand complexes compound. I hope everybody is good. Chalo, perfect. Now, great. Moving to the next one. Again, beta, a logical, a conceptual beauty from the chapter of chemical kinetics, a conceptual beauty from the chapter of chemical kinetics. Now, what is that beauty? What is that beauty? Now, the beauty says you have to consider the first order reaction. R is giving you P. The fraction of molecules decomposed in the given first order reaction can be expressed as, sir, if I'm following a first order kinetics, then definitely I can say K is equal to, or rather KT is equal to ln of initial concentration upon concentration at time t, because all the options are giving you exponential term and you are supposed to give the fraction of molecules decomposed. That means I am focused on this value. So rather you use the other form which says AT is equal to initial concentration into exponential of minus KT. Now what will be the ratio, beta? Sir, A naught AT. Sir, AT upon A naught will be, sir, exponential of minus rate constant time of t. And what are we supposed to give? We are supposed to tell the fraction of molecules already decomposed. So how much you have taken initially, sir? A naught. How much is as present at time t, sir? AT. So difference is the molecules that have already been decomposed. So in order to find that difference, sir, obviously this has to be multiplied by, this has to be subtracted by 1. So mathematically, sir, this also has to be subtracted by 1, sir. I hope everybody is getting what I'm saying, because, sir, A naught minus AT, sir, 100 rupees you have taken at time t, let's say 30 rupees is remaining. So how much have decomposed? Sir, 70. How you will get that 70? Sir, 100 minus 30, 70. Upon the initial that you have taken is 1. So I want this in the denominator, then only I will be getting the fraction decomposed upon what I have taken initially. So mathematically, just 1 minus this and 1 minus. So that means our RHS will also be this. So I guess this RHS you are definitely seeing in option daily. So a logically, theoretically, conceptually sound one. Percentage, that's why is less. Chalo, I hope everybody is clear now. 11% only have answered this correctly. I hope everybody is now clear. Moving to the next one. Quick beta. Quick. Guys, clear? Clearer. Clearer. Chalo, great. This is my first revision video. Is there a chance of me scoring 400 by understanding concept? Yes, beta. Yes, Deepa. Definitely yes. Deepa, definitely yes, beta. Now, the empirical formula of Z you have to determine. So what will be the empirical formula of Z? Sir, N-propyl bromide with Wood's reaction. So propyl is this, sir. So I'll be using this as alkyl group and it will be getting dimerized. So 3 carbons and another 3 carbons, that is I'm having hexane as X. So this hexane will react with vanadium pentoxide at elevated temperature. Then this is the direct reagent for aromatization. So first cyclization, then aromatization. 6 member is definitely free from any type of Beer strain. So you will be getting your beautiful benzene. Clear? Now, sir, Cl2 in presence of Lewis acid will do electrophilic substitution, electrophilic aromatic substitution. But at sunlight, sir, extreme condition is happening. So forcefully addition will take place. So addition will form C6H6Cl6. That means the aromaticity is also gone. Hydrogen is also there and chlorine is also there. This is not perchlorobenzene beta. This is C6H6Cl6. So empirical formula will be CHCl, because empirical formula is the simplest formula. And the question it is asking the empirical weight of Z. So it is not asking you the molecular weight, but rather the empirical weight, which is 12 plus 1 plus 35.5, which definitely makes 48.5 as your correct answer. Chalo, I hope everybody is good. A good one, a logical one from organic chemistry. Yes. Rather, this one I feel, because a question from biomolecules and you are biology students. So I hope this one will be quite doable. The question is for disaccharides. Sucrose is a disaccharide, yeah, definitely, sir. It is having that glycosidic linkage, and that glycosidic linkage is between the first carbon of alpha D glucose and the fourth carbon of the beta D fructose. So this will be alpha D first carbon, and beta D fructose second carbon, sir, the anomeric carbon. So beta D the second carbon. That means, sir, that this goes with first alpha D glucose first carbon and beta D fructose the second carbon. Between them the glycosidic linkage exists. Perfect, sir. That means A is 3, sir. Now what is maltose? Sir, maltose is 1 and 4 of both alpha D glucose. So that means this is 1. What is lactose? Lactose is, sir, beta D galactose and alpha D glucose. So first and fourth carbon. That means C is 4. Then this definitely mean amylopectin is a highly complex branch of all alpha D glucose. The linear polymerization happens with C1 and C4, and the branching happens at first and sixth carbon. So that constitutes your amylopectin. Amylose is just the linear chain. So that definitely means option 1 is your beautifully correct answer. Yes, I knew this. 74% of you have done it correctly. Moving to the next one. That at 25 degrees Celsius, Ka. So this is a weak acid whose Ka is given, and you are supposed to calculate the Kb value of the conjugate base. So this is not valid for all acid base, but yes, sir, for conjugate acid base theory we know, sir. For conjugate acid base theory we know, sir. RE-NEET questions. I have some. So for everyone in the class, chalo, perfect. Now, so sir, for conjugate acid base, sir, we know one thing, that Ka of the acid and Kb of the conjugate base will always be equal to Kw. Please remember this conclusion, beta. So this Kb is nothing but, sir, 10 to the power minus 14, which is Kw of water, upon the dissociation constant of acid, which is 1.8 into 10 to the power minus 4. That means that this is clearly 10 raised to the power minus 10. And here I will be having 1.8. Obviously, this thing will be obviously, okay, just increase one more 10. So one more 10 will make it 10 to the power minus 7. So 10 by 1 by 8 is 5 point something. So definitely option C is your right answer. Chalo, 70% have got it, cracked it. So great. Moving to the next one. The question is in front of you, and I guess you are also quite confident with your chapter of amines, because beta, for amines, even JEE Mains has a high weightage of amines chapter. So sir, NTA. So for your NEET, you will definitely be getting 2 questions from these basic reactions of amines. The NaNO2 and HCl does diazotization. So this guy will be turned to N2 plus and Cl minus as X. Sir, post HBF4, you will be getting benzene diazonium fluoroborate, because that salt is actually stable till room temperature. So this Y is my benzene diazonium fluoroborate. So this CH3 is here, sir. This will be N2 plus, sir. This Cl minus and H plus gone, so it will be BF4 minus. If you simply heat it, it will be fluorobenzene. But I am now heating it with NaNO2 in presence of copper as catalyst. That means I am going to introduce NO2 plus, and that NO2 the NO2 minus, I'm sorry, that NO2 minus will be entering here, because now nitrogen has gone, creating positivity here. So that NO2 minus will be creating a nitrobenzene substituted hydrocarbon. Now this is my product. Now the question is not asking me the product. This is bad. It is asking me percentage of carbon. This is what NTA has done in JEE mains, beta. This is exactly what NTA has done in JEE mains. Now actually you have to give percentage of carbon. So percentage of carbon will be weight of carbon. How many carbons I have? So 6 and this one 7. So 7 into 14. 7 into 12 upon weight of organic compound. So I guess weight of organic compound, if you calculate, yes, you have to be calculative with this. So 7 carbons are, then hydrogen will be 1, 2, 3, 4, 4 and 3, 7 hydrogen. So this plus 7 plus nitrogen is 14 plus 16 into 2 is 32. So that molar mass you have to calculate into 100. So you have to be calculative. Please do this as your homework, and definitely few of you must have solved it and got around 60% as your answer. So great, great. But yes, beta, NTA has done this mathematification of organic chemistry. So I am not saying, because sir, those JEE people, they have 25 questions per test, per chapter, per paper. Sir, we have 45, so we have do not have such so much of time. Sir, I agree, don't attempt such questions in the first go, but yes, there will be 1, 2, 10, 20 students who will be doing them in the last go. So yes, for that last go, and for having that confidence that yes, you can do such questions in your second attempt, not in next year, you should attempt your paper in 2 phases. First phase 1 hour 30 minute, 1 hour 40 minute, and then go for round 2, and these questions you should pick for those round 2, है ना? Hello, great and awesome. Hello, next one, sir. What is the time period of revolution of an electron in the 4th orbit of helium plus? Sir, time period? Yes. Sir, how to calculate time period? Beta, common sense. Beta, time, what is speed? Sir, speed is equal to distance upon time. So what is time? Sir, because speed is equal to distance upon time, so time will be equal to distance. How much a distance electron cover while doing that circular motion according to Bohr model, sir, 2 pi r. And what is the speed? Sir, velocity. So can you calculate 2 pi, what is radius of nth orbit? Sir, r naught into n square upon z. Sir, what is value of velocity of electron in nth orbit? Beta, it is v naught into z upon n. Now, first let us calculate this for whom? Sir, you have to calculate this for helium plus. That means this time period will be 2 pi. What is R naught? Sir, R naught is 0.529 angstrom, as in 10 raised to the power minus 10 meters. I'm putting everything in SI unit because I will be then converting, because I need things in seconds, so everything in SI unit. Upon, what is this constant V naught? Sir, it is 2.18 into 10 raised to the power 6 meter per second. So everything in SI unit. Now what about the constant, sir? So constants is n square upon z, and this z by n. So this is n by z. That means that this is n cube upon z square. What orbit are we talking of? Sir, 4th Bohr orbit. So 4 squared. What nucleus we are talking of? Helium. So 2 squared. The 2 squared is 4. 4 squared is 16. So 16 by 4 is nothing but 4. Because this is 4 squared, na? Yes. Nahi yaar, 4 power, I'm sorry beta. Because n is cubed, sir. So 4 is cubed. So 4 into 16, let me do this. This is definitely n squared. So this is definitely 4. So this 4 and this 4 is gone. So I'll be having 16 over here. So this 16, once you do, I guess you will be getting somewhere around 2.4 or 2.5 into 10 raised to the power minus 15 seconds. And what is 10 raised to the power minus 15? Are those conversions: deci, centi, milli, micro, nano, pico, femto. Femto is the 7th finger, sir. So minus 1, minus 2, minus 3, minus 6, minus 9, minus 12, and minus 15. That means, sir, this is femtosecond, sir. That means, sir, option Delhi is my probable answer, sir. Yes, you have to be on your toes for attempting such questions? Yes, definitely not a question to be attempted on the first go, but yes, not a question which you cannot attempt. Yes, in the second go, after clearing all your easy and moderate level of questions, such questions are definitely going to make the difference. Take your rank, boost your rank to a higher extent, but yes, not for the first part of your paper. So that you have to keep in your mind, beta. I've been continuously mentioning this, that you have to attempt your physics, you have to attempt your physical chemistry per se, not in the first go, definitely not in the first go. Again, high probable logic from ionic equilibrium, the common ion effect. What does this common ion effect tell you? So the pH at which this magnesium hydroxide begins to precipitate from a solution containing 0.1 molar of magnesium ion. So magnesium ion I am creating from outside. This guy, which is already a sparingly soluble salt because its K value is very less, is giving very minimum amount of magnesium ions and hydroxide ions. Because I've created magnesium ions from outside, so this dissociation will be further suppressed, but still some of palm part will be getting dissociated, so S and 2S. What will never change, sir? K value will never change, which is given as 10 to the power minus 11, which will be equal to magnesium ion concentration, which will be this plus this. Obviously, this is much more. So magnitude wise, magnesium ion concentration is just that value, because 0.1 plus S will be approximately 0.1 only. This has nothing to be added. Mathematically, this is irrelevant. And then I have 2S concentration squared. So that's why I'll be getting the value of S, which is this is 10 to the power minus 1. So this becomes 10 to the power minus 10. Under root will become 10 to the power minus 5. So 2S value is this. That means S value is 1 by 2 into 10 to the power minus 5. That means I know OH minus ion concentration. So if I know OH minus ion concentration, POH will be minus of log of this value, which will be giving you the value of POH. So minus of log of this will be 5 minus log 2. Minus log 2 will be, this will be half, na? Yes. So OH minus is 2S. Okay, so 2S value is 10 to the power minus 10. Yes, S is 10 to the power minus 5 by 2. Okay. And after this, 1 by 2. So this is minus of log of this is 5 minus log 10. 5 minus log of 1 by 2, as in 5 log 2. So this will be 5.3. So this is my value of POH. So pH will be, sir, beta pH will be, so pH will be 14 minus 5.3. So 14 minus 5 is 9. 9, this will be 8.7. So I have A also, B also. Beta, just check the calculation. Guys, just check the calculation. Acha, beta, this S I have calculated properly, but is S my OH minus ion concentration? No, sir. Your OH minus ion concentration is 2S. Or rather, why I'm writing this? Why you are unnecessarily writing S? Why, sir? Why should we should not write S? Because beta, you are only interested in OH minus ion concentration. And technically, this magnesium ion concentration into OH minus ion concentration squared will definitely give you the clear OH minus ion concentration. So why bother? Sir, OH minus ion concentration will be 10 to the power minus 10 under root. So it will be directly 10 to the power minus 5. So POH is directly log minus of log of this. That means this is directly 5. If this is directly 5, then I have to just subtract 5. So 14 minus 5 will definitely give you easily and only 9. Good one, beta. Good one. I mean, yes, technically this is a basic thing, but yes, sometimes in your while doing such questions in the exams you tend to make such the more you practice, the less possibility and probability it will become to make such errors. I hope everybody is clear. I hope everybody is clear. Chalo, perfect. Moving to the next one. Time starts in 3, 2, 1. Go quick, quick, everybody, quick, everybody, quick, everybody. Roshan, sir, can you beta see for first of all, magnesium ion was given in the previous question, and because magnesium ion was given, that means that clearly magnesium ion concentration will be taken from the strong electrolyte. Now, for OH minus ion concentration, yes, it was 2s, but ultimately I just needed that pH value. So what we have committed error over there is we calculated S and we thought S is, sir, 2s was the concentration of OH minus ion concentration. So because I'm saying you could definitely write OH minus ion concentration as 2s, then that half will be multiplied by 2. But sir, why we bother? Because in that question we needed the pH at which PPT will start, and the pH, sir, pH will say 14 minus pH, and pH is OH minus ion concentration minus log. So rather just make it OH minus ion concentration. Don't bother for such cases writing S and 2s in the stoichiometry. Eh? क्या let me read that. Let me read that. Chem is chem is try. Okay. Chemistry try किए बिना नहीं आता because chem is try. Yes. Great. Great word editing बेटा. हाँ बेटा. ओरेवा ओरेवा dope. I beta read your comment by after going to my system because I wanted to chemist try. Yes. Chalo beta. Now this is, I guess, a doable one for everybody. 57%. I was expecting more beta. In biomolecules you read in your biology also. Statement 1 is lysine and arginine are essential and basic. Definitely they have NH2 and they are essential. They have to be consumed from outside your body. Do not synthesize them. So definitely first is correct. Leucine and phenyl alanine are non-essential. They are definitely neutral, but they are also essential. For non-essential, I have given you a key. For non-essential, you should remember central. You should be centralized at Gaurav Singh Arora. So CGSA pro-tyro. All the alpha amino acids who are starting with an initial CGSA pro and tyro are non-essential. That means they are synthesized in your body. They are non-essential. You do not have to eat in your diet. They will be given to you. But yes, there is one alpha amino acid which is starting from A, which is essential. So how to remember this? Argentina zaruri hai because Arg9 is an alpha amino acid which is rather essential. So I hope this rhyme will help you. That makes option 1 as your right answer. Moving to the next one, a very doable one, I guess. I guess very doable one. H2O2 reduces KMNO4 in acidic medium to X. So in acidic medium, H2O2, in acidic medium, H2O2 will be oxidized. Sir, oxygen in -1 oxidation state can only be oxidized once you increase the oxidation state, which will be done in case of oxygen. And KMNO4, as in MnO4 minus, will reduce itself in acidic medium to Mn plus 2. That means this X is Mn plus 2. This X is Mn plus 2. So only one option you have. But yes, in neutral medium, definitely this is not neutral, but rather mild basic. So in mild basic and neutral medium, this manganese changes to MnO2. In highly alkaline or basic medium, it changes to permanganate, changes to manganate ion, which is MnO4 2 minus. A beautiful D block logic linked with redox logic. So I hope everybody is clear now. Great, 72%. Moving to the next one. The number of isomers possible for a dibromo derivative. Molecular weight is given of an alkene is. Okay, so dibromo, that means 2 bromine I need. That means 160 That means molecular weight of the compound is this dibromo derivative. 1 bromine is 80, so dibromo will be 180 into 2 as in 160. So 160 if I subtract, I will be getting 6, 2. So 26. So how many carbons I have? Sir, you can just have 2 carbons, sir. 2 carbons. So 12 2 ja 24. Let's say bromine. So 160 is done and remaining 4 hydrogens, sir. Yes. So what are the possible isomers? Sir, one is definitely this. And no one is saying you should take it at the vicinal position. So rather, if I take geminal one, acha, it should be an alkene. So how come molecular mass? Because I have just 2 hydrogens. So yes, my bad. I can just have 2 hydrogens. That means it has to be an alkene. So if it is an alkene, sir, then this is CH double bond CH. Then molecular mass is perfect, sir. And now, okay, sir, now this can show GI, sir. This can show GI. Bromine trans and bromine cis. So one trans, one cis, so 2 isomers are these. And if I'm writing this alkene as geminal dibromide, then this will not show GI, but this will itself be an isomer. So Bombay is your answer. Great question. I mean, yes, logically, if you're trying to do such questions with limited time, so they become great questions because you have to do plus minus also, molecular mass, molecular formula identification also, and that can only be done with plus minus. And then you have to think about organic chemistry. So a good one. I hope everybody is clear. I hope everybody is clear. Guys, beta, whoever is listening, whoever is listening, at least give your teacher that level of extra motivation. Maja aa gaya, sir. Why? Great, sir. Oh, awesome, sir. My JEE people do it. Maja aa gaya, sir. Great, sir. So I hope everybody is good. I need whoever is getting it. Yes, sir. Understood, sir. Great, sir. I mean, whoever is doing questions properly and enjoying the discussion also, or else I can come and just give the. If I'm putting in more efforts, I need appreciation. Chalo. हाँ मजा आ गया yeah great परिमाला ओरे वैडोपे परिमाला विश्व ओके शोएब रेवती world all clear perfect chalo great guys great moving to the next one I guess this is a simple one but yes a rather a logical one. The first ionization enthalpy of element at extreme left, sir, you will be metal, so that will be obviously lowest or minimum. And the negative electron gain enthalpy, yes, sir, electron gain enthalpy also ionization also as size decreases, so it increases, so that means it is highest. So a doable one, sir. C is my answer, sir. People must be reading the question, but yes, now I hope everybody know it. A doable one. You have to consider the reaction now, beta. A high probable logic from chemical equilibrium, characteristic of equilibrium constant K. I have to decide K for this reaction, which is K3. How can I calculate K3? Sir, I want XYZ in the product side. So I just have to multiply K2 because I need this. So just I have to add this reaction, second reaction. So if you just add second reaction, sir, if you add 2 reactions, equilibrium constant gets multiplied. So this K2 will be multiplied by what? Sir, this X2, Y2, I need on left hand side, but with a coefficient half. So first of all, this equation has to be multiplied by half and then it has to be reversed. So because I have to multiply it by half, that means I have to, if you multiply by any value n, the coefficient goes to power of the value of k. and then I have to reverse this also. That means it has to be multiplied by -1, so rather this will be -12. And after that you have to add, then that means these two has to be get multiplied. So just do this mathematical operation. K2 is 5 into 10 raised to the power -3. K1 is this. So first of all, in reverse this value, so this will be 1 upon 2.5 into 10 raised to the power 5, and I have to take under root of this. So because it is 1 by 2. So first I have inverted this value, so 1 by k, so 1 by k, and then I have to make under root. So this will be also easy because this will be 25 into 10 is power 6. So this is 5 into 10 is power -3, and here I will be having in the denominator 5 into 10 raised to power +3. So this -3 and +3 will become -6, and definitely 5 is gone. So my answer is acha, okay, my bad beta, my bad. What my bad? Beta, this was 2.5 into 10 raised to power 5 na. So because I am removing this decimal, so this will become 10 is power 4 after writing 25. So this will be 10 is power 2. So this will be 10 is power -5. That means option C becomes your right answer. Yes. Let me check. Percentage is less. I hope everybody is good. A good one. Mathematically challenging and conceptually sound. Mathematically challenging and conceptually sound. I hope everybody is good. I hope everybody is good. Guys, क्या हाल है sir? Super संयम. I'm good beta. I hope you're good too. Chalo. I'm good. Chalo. Perfect. Once more. Yes sir. Great. Chalo. मजा आ गया. Now this is working. Great, 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 great. Now let us attempt this next one beta. Sir, an excess of silver nitrate is added to 100 ml of 0.05 molar. So how much moles I'm using? Sir, 100 millimoles. So let us calculate millimoles rather. So how many millimoles I'm having? Sir, 100 into 0.05, as in sir 5 millimoles of tetra aqua dichlorido. So tetra aqua dichlorido chromium chloride. The number of moles. Sir, this is having only one chlorine as counter ion. So 1 mole will give 1 Cl. So 5 millimoles will give 5 millimoles of chlorine, or rather AgCl. So 5 millimoles is your answer. You do not have to move your pen for such questions. But yes, a highly logical, a highly probable question from the chapter of coordination complexes. I hope everybody is clear. Moving to the next one. Quick, everybody. I hope everybody is clear. Great, great and awesome. Great and awesome. Now, chalo, perfect, perfect. Yes. Now, consider the following reactions. Total number of electrons in this shall be pi bonds, and lone pair of electrons in the product. So this is my glucose with Hi. Sir, Hi in heating acts as a reducing agent, so it will turn this into hexane. Then sir, aromatization will turn into benzene. Then sir, Friedel-Crafts acylation, but with benzoyl chloride will turn this, because I am using benzoyl chloride in presence of Lewis acid. So Friedel-Crafts acylation, but this will rather be benzoylation. So this is your product. Now the benzene will be having 3 pi clouds, sir. 3 pi electrons. So 3 and 3, 6. 6 and this one is 7. So pi bonds will be 7. Lone pair will only be on oxygen, and how many lone pair on oxygen you will be having? So 2 lone pair. So 2 plus 7 is 9 pair, but you are asked number of electrons. So 9 pair into 2 as in 18 electrons you will be having. Good one. Good one, beta. This one is also good. Percentage is less. I guess people have answered 46. I don't know why. Maybe because of this typo error, but initially only I've told you that is pi. I hope everybody is good. I hope everybody is good. Sir, your teaching is so good. Thank you, Divyanshu. Please spread the word and make use of this for yourself. Chalo. Sir, आज कितना पढ़ाएंगे? Beta, I'll be just finishing around 2 and then you'll be having your physics. मजा आ गया sir. Great, great, great. Moving to the next one. Moving to the next one. Quick. हाँ बेटा, again a question which require your inorganic knowledge and your English, your reading ability. So please read the question properly and do it. Chalo, now. Great, great. हाँ बेटा, this is more than enough, Urvashi, but yes, you have to practice more and more, more and more. So the group 15 element forms d pi bond. So you have to tell what is that group 15 element which is capable of forming d pi among with transition metals. It also forms hydride, which is strongest base. Hai sir? D pi. So that means it can't be nitrogen because nitrogen does not have D. But sir, for if you're talking of hydrides, then sir, ammonia, the lone pair exists in sp3 hybridized orbital. Sir, after phosphorus and the other the hydrides, sir, this lone pair exists in pure s orbital. Why? Because they do not even get hybridized, sir. They do not even get hybridized. Drago's rule. That means S character here is 25% and S character here is 100%. Sir, more be the S character, more is the electronegativity, sir. More be the S character, more is the electronegativity, sir. That means, sir, this is as not as nitrogen, as a molecule ammonia, the lone pair on nitrogen is present in a orbital which is having relatively less electronegativity. So that's why it can be easily donated. That's why ammonia is a best base, best hydride among group 15 hydrides. And the basic nature further decreases down the group. So if you just leave nitrogen, who is the best base? Sir, ammonia. Sir, phosphine, sir. And phosphine can do d pi also because it has D. That means you are talking of phosphorus, which is having atomic number 15. So a beautiful question from inorganic chemistry. A beauty from inorganic chemistry. A beauty from inorganic chemistry. I hope everybody, Gayatri, Kanwan, Abid, then Kirthana. Perfect, beta. Awesome. Awesome. Great. Moving to the next one. Now this is, I guess, a doable one. You have to again read. Beta, now select incorrect and then correct. So please make sure you are reading the peculiarities of NTA properly because they do it and that's why we have framed it. So here you have to select incorrect. Now some student don't do this. Sir, correct is the pucha tha. They asked correct only. No, beta, please read the question. Use your eyes because they are going to challenge your eyes like anything. A calm mind can only be a great observer, beta. So please make sure. Sir, should we stop reading after NEET or continue for another 12 days? Hey? Nahi beta. I hope such things is not going to happen. So rest assured. Don't trust anybody. Trust Mother Nature. Trust the universe. It is not going to happen. Else, I am not saying to trust any XYZ. Trust yourself. Trust the energy of the nature because that is inevitable. and you will definitely be awesome in whatever will be happening after that. Whether you are going to do great or not great, because whatever is going to happen for your good. If you just believe this, if you just actually believe this, that whatever is going to happen tomorrow is going to happen only to make you happy, because it is actually the thing. If you just give that frequency to the universe, it is going to reciprocate. Chalo. Uh, chalo. Now, sir, here the question is, this is a stronger base than yes, sir. Here lone pair is delocalized, sir. That is an alkylamine. So this is definitely correct. So correct, that means not an answer. Now, this can be synthesized, sir. Gabriel synthesized thelimide can only form 1 degree amine, sir. That too aliphatic, sir. This is aromatic, sir. It can't be formed. So because it is incorrect, so an answer. So definitely I will be needing B, and definitely my answer is done because A is not there. So definitely my job is done. Definitely my job is done. But yes, because we are practicing, so that's why let us read through them also. Sir, here Hoffman bromamide degradation will remove this, and then I'll be having benzylic amine, sir, which is primary aliphatic amine, sir. That means this is also incorrect. So an answer, because this is incorrect, because it will be aliphatic. It is saying aromatic. Definitely C is incorrect. And with the diazotization, this will be N2 plus and Cl minus, and after heating under water, so this will be gone and phenol will be produced. So will phenol dissolve in NaOH? Yes, sir. Phenol is a strong acid than water, and it will definitely create salt. So a doable one. So this is definitely correct. So because it is correct, hence not an answer. Moving to the next one. Quick. 3, 2, 1, go. Quick, everybody. Quick, super quick, everyone. The number of carboxylic acid group in the product P. This is my product P, and how many carboxylic acid group I have. So first I have to do acidic hydrolysis, or acidic hydrolysis will first convert this anhydride into carboxylic acid. So this will rather be, sir, a cyclohexene connected with again 6-membered ring having carbonyl group, and this guy will now be C double bond OH. So this is a carboxylic acid, and this is also a carboxylic acid. Now, because I have heated this particular carboxylic acid, then obviously, sir, we should, and everybody should know, after heating carboxylic acid, what is released? Sir, it releases, what is released? Sir, it releases, guys, sir, this is carboxylic acid alpha carbon beta carbon. This is carboxylic acid alpha carbon beta carbon. And what is heating effect of beta keto carboxylic acid? Sir, beta keto carboxylic acid, in order to stabilize, they decarboxylate. So beta keto carboxylic acid tends to decarboxylate, sir. That means they are gone, sir. So this pi is here. This is double bonded O, this is double bonded O, because they will decarboxylate, sir. So this will become CH and this will become CH. I hope everybody's with me. That means after this, I have to do ozonolysis. Sir, ozonolysis of alkene will do what? Beta ozonolysis of alkene will break this double bond and will add double bond O. So ultimately here I am having, here this part will be saturated now. But now this part will be opened up. This is carbon, and this carbon is attached with a this alkene carbon, and this carbon has just now been introduced with carbonyl group. But because this is oxidative ozonolysis, so this will also become, the H will also become OH. So how many carboxylic, how many COOH group I am having? Sir, you are having 2 COOH group. But yes, rather a good question, beta. Rather a good question, employing the heating effect of carboxylic. What is the rule that you should know? Sir, beta keto carboxylic acid tends to decarboxylate. So remove CO2, form the corresponding alcohol. I hope everybody is good. I hope everybody is good. Chalo, perfect. Moving to the next one. Again, a beauty from chemical bonding beta. Boron trifluoride, as in BF3, auto ionizes. I mean, two molecules will be utilized. One has lost fluorine as F minus, one has gained. So this is BrF4 minus. Now the shapes. Sir, this bromine is definitely forming two bonds. One electron is gone, so 6 will be remaining in bromine. 6, sir, 2 gone. That means a 2 lone pair. So 2 bond pair, 2 lone pair, as in this Br plus will be just like water. So this will be a bend shaped molecule. So this will be a bend shaped molecule, not linear. Bend shape. And what about this BrF4 minus, a bromine getting extra electrons? So 7 plus 1 is 8. 4 bond pairs are, so 2 lone pair. So obviously those 2 lone pair will be aligned opposite to each other, because this will be AB6 type of molecule. So the 4 fluorines will be coming in the single plane. So this is nothing but square planar. I hope everybody have got this one. 72%. Great. Great and awesome. Moving to the next one. Time starts in 3, 2, 1, go. So you have to complete the following reaction sequence and give the name of major product. So, complete the following. What is that reaction sequence? Acha. So I guess that's a misprint. Complete the following. Where is the reaction sequence? So that's a misprint, beta. So rather, in the same time, do this one. That's a typo error, beta. There were no sequence. So rather attempt this one. Quick. 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 Yes. Chalo. Great. The question says, beta, for such questions now, always go for options, because if you go for options, you will be clearly getting your answer. You will be clearly getting your answer once you just go to the options. Like for example, over here, A is either 4 or 3. Sir, 3 options are saying 3, is obviously don't check A first. Sir, B is either 2, 4, 3. 2 options are 4, so maybe. C is either 2 or 1, and D is either. So just check, I mean, yes, just check CD. That will rather give you an answer. So C is 1.8 mg carbon, and you have to give number of atoms. So carbon is definitely, sir, atomicity is 1. So number of moles will be 1.8 into 10 is power minus 3 upon 12. And sir, number of moles is number of species into Avogadro number, which is 6 into 10 is power 23, or rather, because everywhere Na is written, so don't write this. This is Na. Now this is just multiplication, sir. Sir, this is 6 2 jar, 6 3 jar. That means this is 0.3. That means, sir, this is 3 by 20 into 10 is power minus 3, or rather 10 is power minus 4 with 3 by 2. So this is 1.5 into 10 is power minus 4. So if I'm having 1.5 into 10 is power minus 4, that means C is definitely 2. So if C is 2, that means we have our answer, because none of the option is saying C2. So yes, if you go by options, maybe you can save a lot of time, because formula basic is nothing. So number of moles is equal to weight in gram upon molar mass, and it is also equal to number of species upon Avogadro's number. But yes, most of the time you will be getting such things. Make sure you are having a handsome eye on the options first to decode the question quickly. है ना? Chalo. Moving to the next one. Time starts in 3, 2, 1, go. Yes, a good one from or practical organic chemistry beta. And I guess this will be the second last question for us. Acha. Let me just check how many questions we have. Chalo, do this one, beta. By the time I just check Subrat sir whether he's ready or not. Chalo, I guess we just finish these questions first because I would just want to finish these questions. High probable logic beta. Chalo. So I guess most of you have solved it. The answer to this one is obviously Bombay. Now why? Percentage is less, so definitely you require understanding for this particular question. Sir, this is talking of thin layer chromatography. So again, sir, a purification and separation technique. A basic and a beautiful logic, not from anywhere else, from NCERT GOC, General Organic Chemistry, class 11th. The distance of spot A and B are 5 centimeter and 7 centimeter. So this guy, this line is 5 centimeter. This is 5 centimeter and this is 7 centimeter. B is rather 7 centimeter from the baseline. From the bottom of a TLC plate. Technically, beta, in your TLC plate, you have a baseline. This is my baseline. And we generally calculate with respect to the baseline because solvent will move more, because solvent will have more moving tendency and solute will move less. Which solute will move to the lowest possible extent, which is having more rate of adsorption. So definitely A is having more rate of adsorption, hence A stopped here, B has rather moved here, and solvent has rather moved till this point. So technically, from the baseline, because it is 1 centimeter here, 1 centimeter here, and this complete thing is 1 centimeter. So, sir, how much solvent has moved? Sir, from here till this point, which is 1 centimeter below from the top. That means a solvent has moved 8 points. Because I will be measuring the movement from the baseline. So baseline is 1 centimeter top, that is 1 centimeter below where solvent is reaching. So out of 10, 1 and 1 gone. So that means solvent has moved 8 points, 8 centimeters. So because A has moved 5 centimeters, as in sir, this difference is nothing but 4 units, sir. And because it has moved 7, so from the baseline, movement of A is nothing but 6 centimeters. Now, what is my retardation factor? So, retardation factor as per NCERT is the distance moved by species, any species which you are separating, upon distance traveled or moved by solvent. Sir, but solvent will be same. Yes, I'm not denying to the facts, but still I'm just making you revise everything properly. So what is retardation factor of A? How much A has moved? Sir, 4 centimeter. How much solvent has moved? Sir, 8 centimeters. So retardation factor for B will be how much B has moved? Sir, 6 centimeter. How much A solvent has moved? Sir, 8. Now what you have to calculate? You are saying RF value of B is this much times RF value of A. That means you have to find the ratio retardation factor of B upon retardation factor of A. So definitely of B will be 6 by 8. So this is nothing but 6 by 8. This is nothing but 4 by 8. That means this is 6 by 4. That means this is 3 by 2. That means this is 1.5. Because you have to report as x into 10 raised to power minus 1. So this 1.5 will be 15 into 10 raised to power minus 1. That means answer is obviously Bombay. A good one, but an application of NCERT beta. A beautiful application of NCERT. Please make sure. The reason I'm bringing you these good questions is because I am moving along NCERT. है ना? Great. Chalo, perfect. Perfecto. Moving to the next one. Time starts now. Quick. 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 मजा आ गया. Great. चलो. मजा आ गया सर. Great बेटा. चलो. Now what does the question tell you? First one is methane can be prepared by decarboxylation of sodium ethanoate using coal-based electrolysis of sodium acetate. है? अच्छा, then I have a comma. Okay, I'm sorry. Beta, make sure you are reading question properly. Na? Methane can be prepared by soda lime decarboxylation. Yes. How? You do soda lime decarboxylation of sodium salt of ethanoic acid. So this will be removed and this methane, this can create methane. Yes. So this is true, sir. This part is true. Kolbe electrolysis of sodium acetate. No, sir. If I do Kolbe electrolysis of this, if I just do electrolysis, then, sir, CO2 will definitely go, but it will create a radical, sir. And this radical and this another radical will do chain termination. So minimum ethane will be formed, sir. Methane, this is false, sir. The first part is true, rather. And reaction of CH3, yes, with water, Grignard agent. So this is CH3 minus, it will snatch H plus of water, and this can also create methane. So out of 3 statement, 2 are correct, sir. But in between, with a comma, they are doing bad things, sir. So definitely because of the middle thing, it is incorrect. And that's why statement 1 is definitely complete statement becomes incorrect because of the middle portion. Now, methane cannot be prepared from unsaturated hydrocarbons, yes, and by Wood's reaction, definitely correct. So statement 2 is perfect. So statement 2 is perfect, but first one is incorrect because of the middle portion. I hope everybody is good. A good question, beta. A good question, a direct conceptual understanding of how NTA is going to trick you by writing theory, by making you anxious that or we have to read so much, and how you will get a habit of reading by practicing more and more, rather not getting that feeling of anxiousness, but rather getting your confidence by reading more and more. So read questions. The more and more you read, the more and more you will be not facing any set of emotional challenge, and the more calm your mind will be. So even if you are having any feeling while feeling that level of extreme emotions, do some work. What work? Attempt. So that's why we will be keeping these lives really active. I will be coming live tomorrow also. That too, I will be starting at 6 p.m. So 6 a.m. in the morning. So make sure you set yourself completely and join us tomorrow also, live at 6 p.m. morning, early morning. So as to share that energy. Anna? Chalo, perfect. Now, logical one from thermodynamics, you are talking of isothermal processes. So for isothermal process, a temperature is constant, and because internal energy is a function of temperature, so change in internal energy will definitely be 0. So as per first law, so delta U is equal to Q plus W. Because this is 0, that means whatever heat you will be giving, it will be work converted to minus of W. Now, what can you say about reversible expansion? Sir, reversible expansion, okay. So because you are expanding, sir, that means work is done by the system. Because you are expanding, that is work is done by the system. Okay, sir. That means this W will be negative, and this negative will make Q positive, sir. Sir, what will be the value, sir? Beta, what is reversible work? Sir, reversible work will be minus P external. So minus will become plus. Minus the external pressure into, or rather this is not irreversible, so plus nRT ln of final volume upon initial volume minus P external dV is when external pressure is constant. When it is constant, sir, during irreversible work. So here ocean is asking us reversible, so obviously it will be minus nRT ln of V2 by V1. That means where you have that, so here that means, sir, A is definitely 2. That means A is 2, so only one option is gone. Now free expansion, so free expansion as in you have no external pressure, so that means that work is 0, so Q is 0. That means definitely B is 1, sir. So B1, so this gone. Now we just have to check C, either 3 or 4. So what about C? So C is saying irreversible compression. Now because it is compression, sir, and irreversible, so external pressure will be constant, so minus P external to delta B will be your work. That means, sir, C is definitely 3. That means C is your answer. And for cyclic process, we know for cyclic process, for cyclic process, you can clearly have Q reversible upon T, heat exchange between system and surrounding upon T, that is delta S. Sir, for cyclic process, all the changes, thermodynamic changes, because this is nothing but delta S. So because it is, sir, they are state function, and for a cyclic process, all state function will be 0. So definitely this is also correct. Moving to the next one. Again beta, a good one. Why I'm saying good one? Because you have to read through the statements again. You have to read through the statements again, which is rather challenging. And inorganically a good question. Aluminium is more electropositive than thallium, as a standard electrode potential value of E0 is negative, and for thallium plus 3 to thallium is positive. Now beta, technically why I'm saying it is a good question, because if you just go by class 10th of basic, normal basic, you will say down the group metallic nature increases. So sir, instead of aluminium, thallium should be a metal. But beta, it is talking of thallium in +3 oxidation state, and then you should also keep in mind the factor of Fazan's effect, the inert pair effect. Sir, because of inert pair effect, thallium +3 will be highly unstable, sir. Thallium +1 is stable, sir, and it is talking of thallium +3. And definitely, sir, who will want to be in +3 oxidation state, sir? Aluminium, because for thallium +3 is not at all stable, sir. Thallium +3 will love to reduce itself. That's why its reduction potential is also positive. So if a metal wants to reduce itself, that means that it is not a perfect metal. So for thallium, for these because of this inert pair effect, definitely, sir, this is correct. I hope you're getting it now. The sum of first 3 ionization enthalpy of boron is high, very high, when compared to that of aluminum. Yes, sir. According to this is periodicity. That's why we know, sir, that generally these aluminium, they form ionic compound. Why boron form covalent compounds? Because ionization is very high. Boron coming in +3 oxidation state is not very often. So due to this reason, boron forms covalent compound. Definitely correct. So both statements are correct. That means option C is your beautifully P block. Awareness wise, this is a good one. 53%. Great. Moving to, chalo, attempt this one, beta. Attempt this one. Practical application of electrochemistry. Quick. A practical application of electrochemistry. Quick. Now, I hope you are doing it, sir. For these questions, we have to do nothing. 1 mole water where oxygen is in -2 oxidation state is going to 0. So, sir, charge transfer will be, sir, 2 electrons will be transacted, sir. So 1 mole will have 2 mole electron getting transferred. So 1 mole will have 2 mole. That means 2 Faraday charge will be transferred. That means, sir, 1 is definitely 2. That means A is definitely 2. So these 2 options are gone. Then we just check Bombay and we are through. Sir, 1 mole Mn plus 2. Beta, Mn plus 2, if you want, then yes, for that you should definitely know that technically if any manganese is doing any of those reactions beta, then technically what you should have, I guess uh, this should be more clearly. So let me reframe this, because technically this is incorrect beta. MnO4 minus in acidic medium it has to mention, because if it is so, then MnO4 minus in acidic medium will convert to Mn plus 2. Yes. So I guess this is, I mean, if such a question is coming, then they will definitely tell you, because sir, for permanganate it depends upon the medium. Come on, sir. Agreed. So that's why I'm saying it is mentioned. Let's say it is mentioned that you have 1 mole of MnO4 minus in acidic medium. That means, sir, manganese from +7 oxidation state is going to +2 oxidation state. So charge transfer will be 5 electrons for 1 molecule. So for 1 mole molecule, rather it will be 5 Faraday. So this will rather be 5 Faraday, as in Bombay is 4. Bombay is 4. That means A will be your right answer. I hope everybody is clear. I hope everybody is clear. Chalo, great. Now that means, sir, A is my right answer. Great, great beta. Great. Everybody doing super awesome. Now let us attempt this one. Yes, I want you to have an idea of graphs also from atomic structure. By the time, let me just see if Subrat sir is ready. Chalo, maza aaya. Yeah, mast thi class sir. Thank you beta. So the more you practice your content beta, the stronger you feel for yourself. Chalo. So you have to give the variation between probability, density, psi square and the distance of electron from the nucleus. Now beta, such graphs, let me just share a basic about this. Beta, I mean, only technically if you plot graph for s orbitals, then specifically for s orbitals, how shall this graph look? What graph, sir? Beta, graph of probability with distance from the nucleus. Beta, what is shape of s, sir? It is spherical. That means at the center the probability of finding electron is what, sir? It is maximum. That's why for s, graph shall always start from infinity and then it approach 0. And for all other set of orbitals, p, d, f, for all such orbitals, what is their shape? So they always start from 0. P is a dumbbell, so D is double dumbbell, F is diffused, although we do not write their shape, but we should know this, that why we design such electron distribution, because at the center the probability of finding electron is 0. Now that means for them, graph should always, for graph of sigma should always start from 0, and after a node the graph becomes negative, if at all there's a node. Sir, what is a node? Beta, node is a region where probability of finding electron is 0, because I'm moving radially. So what node you should calculate, sir? Radial nodes. Nodes at the center and at infinity is not considered, because at the center and at infinity you should not consider that node, because sir, you should only start from origin. At infinite distance no one will have any impact, so nodes are only counted in the between. Okay, sir. Now what graph I'm having, sir? I'm having graph of psi square. So now you tell me what will be the graph of psi, sir? Because it is squared, so this must be positively, I mean, I'm not talking of magnitude. What will be the graph of psi? So obviously it is starting from maxima, then it is attaining a 0 value, then sir it must be doing like this. So node at even if it was 0 and at infinity it is going to be 0, but we consider only node in between. That means how many radial node it is having, sir? Only one. So definitely the psi is like a wave. So wave can have positive notion also, wave can have negative notion also. This is not charge. So a beautiful application of graphs. So definitely, sir, this is starting from the maxima, that is definitely it is belonging to a s orbital. But what s orbital, what energy level, sir? How many radial nodes it is having? One. So which among them is having one radial node? So radial node for this will be 1 minus 0 minus 1, so radial node will be 0, so no sir. 2 minus 0 minus 1, yes sir, radial node is 1, so definitely this is your answer. That means Bombay gives you your clear cut picture. 80%, great, awesome guys. Moving to the last one. The last question says, in which of the following galvanic cell EMF is maximum? Okay sir, what is the cell, sir? The cathode and anode are same, sir. So let us write the reaction quickly. Sir, magnesium is forming magnesium plus 2. This is the oxidation half. Who is getting reduced? Sir, chlorine is getting reduced. Who is taking that thing, sir, from 0 to -1? Because platinum, chlorine is a gas, so it should be passed in platinum. That's why you put those terminals at the end, irrespective of whatever reaction is taking place. You always put these terminals at the end, irrespective of whether it is showing that it is oxidation half or reduction half. This much you have to know, that if I have salt bridge, this is my oxidation half anode and this is my reduction half, and then you have to judge that reduction oxidation states decreases to 0 to -1. That means the chlorine gas will be reacting and Cl minus will be produced, because you have to balance. So I guess charge and atom, everything is balanced. Now, so this is pure solid, sir, this is gas, and these are ions, sir. Who changes the who disturbs the rate of a reaction are ions and gases. Okay, so what will be reaction quotient? A reaction quotient will be concentration of magnesium ion raised to the power 1 concentration of chloride ion squared upon all pure solid and pure liquid do not change the state of a react or the state of equilibrium, sir. This do not impact the state of equilibrium, so rather gases and ion does. So I should have partial pressure of gas, but what is the stoichiometry? 1, so raised to the power 1. Applying Nernst equation, I can easily see, sir, E cell will be equal to E naught cell, which is constant, because for all the possible options you are having same cathode and same anode. So sir, obviously not, the standard cell potential will be same. Minus, sir, it will be 0.059 upon number of electron transferred, which is also 2 in this case, into log of reaction quotient Q, because at equilibrium the cell will not function. At equilibrium E cell will be 0, but sir, Q will be equal to K. Now my point is, the question is asking me who is having maximum E cell. Sir, if I want this value to be maximum, then I want, so this is constant, so everything is depending upon log. More the value of log, E cell will be reduced. So I want this Q to be less. So E cell can only be maximized once Q value is least. So you do not have to apply formula, you just have to check the value of Q, whichever is having the lowest value of Q. And I guess from the values only you can see this is more 10 raised to the power things. So if I calculate value of Q for last case, yes, you can calculate for all. I'm just making you calculate for last. So for magnesium ion, it is 10 raised to the power -2, power 1. For chloride ion, it is again 10 raised to the power -2, but it has to be squared. And partial pressure is 1, so don't bother. So this will be 10 raised to the power -6. So who is having least Q? Sir, option 4. So who will be having maximum E cell? Sir, option 4. That means what will be your most probable answer? Sir, option 4. Yes, you can definitely calculate Q for all other cases, which I guess most of the students have incorrectly done. And I knew that that type of question technically is quite challenging for students, but yes, I have tried to give the complete explanation, and I hope now that percentage will definitely change. So with that, beta, all the very best. We will be meeting tomorrow also at sharp. We will be meeting tomorrow also at sharp 6 a.m. And till that time, take care of yourself. Bye bye. Over to Subrat sir. After this, you'll be having a physics beta, hai na?
Hi everyone, welcome back. Good afternoon everyone, good afternoon. Quickly let me know in the chat box. Hello. Let's start physics without any delay. Let's start physics, my dear friends. Hopefully all of you are doing fine. Today is Saturday, tomorrow is your RE-NEET exam. All the very best. You have done your best, alright. Focus on your study and believe in your preparation. And right now let's start physics. Okay. So, my dear friends, in physics, first question is this. This is the first question. In the meanwhile, if you have any problem related with last minute revision for physics, do let me know. I'm going to share some tips, all right, for Saturday and for tomorrow morning, right? For now, let's start this problem. In this question, they are saying a uniform magnetic field B exists in a cylindrical region. This is a cylindrical region, all right, of radius 10 centimeter as shown in figure. A uniform wire of length 80 centimeter and resistance 4 ohm is bent into a square. So this wire has got a resistance of 4 ohm and length is of 80 centimeter and is placed with one side along the diameter of the cylindrical region. If the magnetic field increases at a constant rate of 0.010 Tesla per second, find the current induced in the wire. Let's start. 60 seconds. Let's begin. You should know when they are talking about induced current, we have to use minus d phi by dt. We have to use minus d phi by dt, my dear friends, Faraday's law. And right now, this we can actually rewrite this thing as B into A, and area will come out. So this will become minus A dB by dt. And kindly note, only this is the area along in which flux is linked, right? So we are going to talk about only this area. We are going to talk about only this area, and this is going to be pi r square divided by 2. This is going to be pi r square divided by 2. Please note, value of pi is 3.14. Square of radius, that means 10 square, and we have got centimeter, so 10 to the power minus 4 whole divided by 2. So when you are going to substitute it over here, this is minus 3.14 into, this will become 10 to the power minus 2, right? This will become 10 to the power minus 2, and then multiplied by, dosto, dB by dt, they have given it as it increases with this rate. So 0.10, 0.010, right? And divided by 2. Find this answer. Quickly let me know in the chat box what answer you are getting from this. Be quick. Let me know what answer you are getting from this. See, this is going to be around 10 to the power. If you are going to solve this, I'll help you in these types of calculation also. This is the last step. This is minus 3.14 divided by 2 multiplied by 10 to the power. This is actually 1, 2. So if we are going to actually put one more 0, we are going to simply get 10 to the power minus 3. So we can write it as 10 to the power minus 3 and minus 2 minus 5. Right? So we are getting something like this. And if you are going to solve this, my dear friends, you are going to get nearly first as the answer. As I can see, this can't 0.2 can't be the answer. This is obviously, this is 0.8 and this is 10. So correct answer is going to be 3.9 times 10 to the power minus 5 volt, and with a negative sign. Negative sign is basically for Lenz's law. So moving on, correct answer is option A. Very good. If you have any doubt, do let me know. Please note that we don't have to do anything with this length, right? We don't have to do anything with this length. Acha, just a second. This is right now, this is simply the voltage. They want us to find out current. So current will be actually, the last step will be, current will be this voltage E divided by resistance. They have also given us the resistance. This will come out to be, the magnitude of this will come out to be 3.9 into 10 to the power minus 5 ampere. Option A is going to be absolutely correct. You don't have to do anything with this, but certainly you will have to use the value of resistance, right? So this has to be used and we have to do nothing with 80 centimeter. Got it? Moving on to the next one. Let's see this question. In this question, they are saying following figure shows the multiple reflections of light ray along the glass corridor where the walls are either parallel or perpendicular to one another. If the angle of incidence at point P is 30 degrees. So at this point, my dear friends, angle of incidence is 30 degree. This is 30 degree. Please note, this angle of incidence is 30 degree. So this will be also 30 degree. Clear? Further, they are saying that if the angle of incidence is 30 degree, what are the angle of incidences of the light ray at points Q? So that means you have to find out angle of incidence at point Q, at point R, then at point S, and we have got 2 points S. Sorry, this is point T. This is point T. So point Q, point R, point S, and point T. Let's start. 60 seconds. Let's begin. Quick. And in the meantime, let me just tell you, if this angle is 30 degree, this angle will be 30 degree. So this angle is going to be 30 degree. So the first answer is going to be 30 degree. So this can't be your answer, right? This is going to be 30 degree. This is going to be 30 degree. Dosto, please note these are simply alternate angles. And if this is 30 degree, please note that this is 90 degree. This is 90 degree. This is going to be 60 degree. So for R, it's going to be 60 degrees. So first 30 and second 60, that means this can't be your answer. Either it's third or it's second. Then this is also 60 degree. And if this is 60 degree, then this is also 60 degree, right? So answer is 30, 60, 60. 30, 60, 60. So this third is the correct answer. D is wrong. And if this is 60 degree, again, this is going to be 30 degree. If you have any doubt, do let me know. Third is the absolutely correct answer for this one. 60 seconds is still not over. Very, very good. Moving on to the next one, my dear friends. Moving on to the next one. Questions are of moderate difficulty level. Please read the question properly, what they are trying to say. In this question, they are saying a metallic square wire of side 10 centimeter and resistance 1 ohm is moved with constant velocity. So this has got resistance. Please note, this loop has got resistance 1 ohm. And it is being moved with a velocity of, constant velocity of V naught. And then they are saying in the magnetic field of induction 2 Tesla. So magnetic field induction is also 2 Tesla. The magnetic field perpendicular to find the loop is connected to a network of resistors, each of value 3 ohm. So each of these resistances are 3 ohm. As we can see, doston, this is a balanced Wheatstone bridge. This looks like a balanced Wheatstone bridge. Balanced Wheatstone bridge, right? And the next thing that we can actually do is we can actually find the R equivalent of that. But hold on, let's read the question completely. The resistance of the lead wires OS and OPQ are negligible. What should be the speed to have a steady current of 1 milliampere? Now please note, first of all, let's find out the R equivalent of this. So R equivalent of this entire Wheatstone bridge is, doston, we can remove this 3 ohm. This 3 plus 3 will become 6. This 3 plus 3 will become 6. 6 and 6 will become 3. So R equivalent of this entire combination is 3 ohm. Got it? Further, my dear friends, please do not forget that the metallic square loop also has got a resistance of 1 ohm. That means net resistance will be resistance due to that loop, that is 3 ohm, and plus resistance due to this square loop, that is 1 ohm. This is going to be 4 ohm. Hopefully this is all clear, right? Now next thing that we are going to do is, next thing we are going to say is that induced EMF or induced current can be written as induced EMF divided by net resistance or equivalent resistance, right? Now they have told us that current is actually 1 milliampere. Current is actually 1 milliampere. So if current is 1 milliampere, my dear friends, from here we can find out E. So E will be 1 milliampere multiplied by R net, and R net comes out to be 4. That means this will come out as 4 millivolt. 4 millivolt, right? And what is the formula for induced EMF, dosto? In these types of cases, do you remember induced EMF is simply BLV. BLV, right? So BLV, magnetic field B is given as 2 Tesla, velocity we want to find out, and length, they have also given us the length, that is 10 centimeter. So 10 multiplied by 10 to the power minus 2, and this should be equals to 4 milliampere. So my dear friends, if you are going to pay attention, this will become minus 1 and this will become 2. So you will get your answer as 2 multiplied by 10 to the power minus 2 meter per second. 2 into 10 to the power minus 2. So this can't be our answer. This can't be our answer. Now we have to also find out the direction. We also have to find out the direction. For direction, my dear friends, please note that this is moving in this direction. Got it? This is moving in this direction. That means if you are going to find out induced current, magnetic field inwards, let's find out the battery direction along this wire. Magnetic field inside, thumb this side, current is moving in upward direction. So if current is moving in upward direction, that means battery is going to look somewhat like this, and current will move like this. So current is going to move like this. So this is, mind you, this is a clockwise direction, right? This is going to be a clockwise direction. So C is the correct answer. C is going to be the correct answer. A very nice problem. I have not given you poll in this one. Please note everything is clear. A very, very nice question. Done. Quickly let me know in the chat box, then I'm going to give you poll for this question. Going to give you poll for this question. Quickly write down in the chat box. Everything is sorted. 2 marks on glass rod 10 centimeter apart are found to increase by a distance of 0.08 mm when the rod is heated from 0 degree Celsius to 100 degree Celsius. A flask made up of the same glass as that of rod measures a volume of 1000 cc at 0 degree Celsius. Volume it measures at 100 degree Celsius will be start. Time starts now. 60 seconds. Let me explain it to you what they are saying. They are saying that 2 marks on a glass rod are 10 centimeter apart, and they found to increase their distance by 0.08 mm. That means delta L is 0.08 mm. 0.08 mm when the rod is heated from 0 degree Celsius to 100 degree Celsius. Dosto, the formula for delta L is, if you remember, it is L alpha delta T, right? Is it clear? Everyone remembers this formula, right? So from here we can find out the value of alpha. Alpha will be 0.08 divided by L. L was, mind you, 10 centimeter, and if you want me to convert everything into SI unit, I can do that. This will become 10 to the power minus 3. L will become 10 centimeter. That means 10 into 10 to the power minus 2 multiplied by change in temperature was delta T was 100. So I'm going to write 100 over here. Hopefully everything is sorted. Now, my dear friends, if you are going to pay attention, this will actually become, let's keep it as alpha. Okay, let's keep it as alpha. If you want to solve this, we can cancel out these two zeros and you can write down this as minus 4. So finally you will get 0.08 into 10 to the power minus 4. Now pay attention, they are talking about volume. They are talking about change in volume. Dosto, what is change in volume? For change in volume, we are going to use the formula as delta V equals to V multiplied by gamma times of delta T. Is it clear? Quickly write down in the chat box if this concept is absolutely clear, and gamma can be written as 3 times of alpha. Gamma can be written as 3 times of alpha. We have found out what alpha is. Let's substitute it. So delta V will be V. Let's not change the unit because they are in the options also they are talking about in cc. V is 1000 cc. So 1000 cc multiplied by 3 times of alpha. Alpha is this. That means 0.08 multiplied by 10 to the power minus 4, and then you have got delta T, and delta T is the volume it measures at 100 degree, and by the way this 1000 was at 0 degree, so for this also delta T is 100. Clear? So my dear friends, 3 zeros, 4 0, we can cancel out this. Hopefully everything is clear till here, right? I have got 10, so I can remove one more point. So 3 into 8 is 24, that means 2.4. 2.4 cc. It will increase by 2.4 cc. That means answer is going to be 1000. So your new volume will be original volume plus delta V. So 1000 plus 2.4, that is option number A. Clear? Everything is good. Problem was nice. Please check it again. Option 1 is absolutely correct. Almost half of you have said option B, but option 1 is absolutely correct. Any doubt, my dear friends? Done? Clear? Can we move on to the next question? Please let me know. Next question is this. Easy question, right? Till now we have discussed, I think, 1, 2, 3, 4. 4 problems. Out of 4 problems, 3 were of moderate difficulty level. This problem is saying, I'm starting the poll also. This problem is saying angle of polarization for any medium is 60 degree. What is going to be the critical angle? What is going to be the critical angle? Simply remember, doston, what do we mean by angle of polarization or polarization angle? See, what happens is, let's suppose a ray is coming, and this is a ray, and this is unpolarized ray. Unpolarized ray, right? It will get incidence. If let's suppose this is the angle of polarization, if angle of incidence is equals to angle of polarization, then the reflected ray will become completely polarized. Completely polarized, and the refracted ray will become partially polarized. Got it? And the next thing is that these are going to be mutually perpendicular. Angle between completely the reflected ray and refracted ray is going to be 90 degree. Got it? And this is basically, this happens when tan of angle of polarization is equals to mu. This is the case. Done? Please remember this. And the next thing that you want to do is, you want to simply write that tan angle of polarization is 60 degree. So that is equals to mu, which means that mu is equals to √3, right? And if now somebody wants to know the critical angle for the same thing, if they want us to find out the critical angle for this medium, we know that critical angle is that angle for which again the refracted ray will make an angle of 90 degree, right? So this means that sine of critical angle will be 1 upon mu, that is 1 upon √3, and IC will become sine inverse 1 upon √3. Sine inverse 1 upon √3 is option number 4th. Clear? Dosto, be quick. Be quick, my dear friends. 1 upon mu, na bache, 1 upon mu. 1 upon mu. Please note how you are going to apply this. Let's say this is IC. So you are going to write sine IC just a second. Have I done some mistake? Is equals to mu times of sine 90 degree. Sorry. My bad, bache. But just a second. Let me check if they have given 60 degree correctly or not. Just a second, dost. No, I think I was correct, na, bache? I was correct, absolutely correct. Please understand this concept. They are saying it, what will be the critical angle for this, right? What will be the critical angle for this? Mu is coming out to be √3. Mu is coming out to be √3, my dear friends, right? Mu is coming out to be √3, and this is, please understand that this is going to be the denser medium, and this is going to be the rare medium. I was absolutely correct. IC is going to be sine inverse 1 by √3. This is absolutely correct. Don't make any mistake. This is going to be absolutely correct. D is correct. Please understand. Very good. TIR will only happen when ray are going to move from denser to rarer. So this option is absolutely correct. Okay. Next. Figure show the position time graph xt graph for one dimensional motion of a body of mass 500 gram. What is the time interval between two consecutive impulses received by the body? Easy problem, 60 seconds. They want us to find out two consecutive. What is the time interval between the two consecutive impulses? Impulse basically means the point where impulse basically means change in linear momentum. That is what impulse means. So let's find out where you are seeing that momentum is changing. At what point you are seeing momentum is changing. Now for an xt curve, the slope of xt curve will give velocity. So the moment velocity changes its direction, at that moment we are going to say that momentum is also changing. So we are seeing that from 0 to 2 seconds velocity was positive, and exactly at 2 seconds velocity became negative. So momentum is getting changed at 2 seconds. Now the next point momentum is getting changed is at 4 seconds. Mind you, it is 4 seconds, not 6 seconds. It is at 4 seconds. So the time duration is actually of 2 seconds. So answer is going to be A. Let me know if you have any doubt. Some of you have thought it's going to be 4 seconds. Some of you have thought it's going to be 6 seconds. Do let me know if you have any problem. We have to find out when the momentum will change, right? So it's something like this, dosto. Let me explain how this problem. It's basically they are talking about, let's suppose a ball is being dropped or thrown. Yeah, it is being dropped. So at this point, it will strike the ground and then it will move to the same height, and then again it will fall, and then again it will move. So at this point, let's say this is t equals to 0. So this is 2 seconds. Then this is, again here we are having some collision, let's suppose, right? Or collision, or it is getting at rest, and then again it is coming back. So 2, 4 and 6 and so on. Hopefully everything is clear. Moving on. In the following diagram, the Wheatstone bridge is balanced when? Chalo, solve this. Solve this, my dear friends. About balanced Wheatstone bridge. Be quick. Come on. Sir, could you draw the VT graph for this? Huh, okay, I can obviously draw the VT graph. I'll draw for you. No problem. I'll draw VT graph for you. Let's solve this and then I'll draw VT graph for you, okay? Be quick. First, let's solve this and then I'll draw the VT graph. Acha, in a balanced Wheatstone bridge, the thing is that whatever we have to do, resistance on AB divided by resistance on AD should be equals to resistance on BC divided by resistance on DC. Right now, this is not the condition, of course, because this is 4 by 18. But what will happen if I'm going to have, if I'll swap these two? If I'll swap these two, see, the most, the easiest way to do this is, let's suppose you have got 4 and you are swapping 6 over here, right? So I think that will make sense, because then you will have 4 upon 6, that is 2 by 3, and you have got 12 divided by 18, that is 6 times 2 and 6 times 3. So this is absolutely correct. So we can swap 18 and 6, right? We can swap 18 and 6, that is option number 4. Option number 4. Very good. Clear. Somebody was asking about VT curve. Definitely I'll draw the VT curve. Let's see. So this is V, this is T. This is positive slope. This is positive slope. This is positive slope. So graph will be drawn something like this. And then suddenly at 2 seconds, the velocity becomes negative. Slope is negative. So that means now the velocity will become negative. And then again, after this 2 seconds, that means 4 seconds, velocity will be over here. And it will keep on changing the direction. Is it fine? Quickly respond. Next question. 20-gram boiling water is poured into an ice cold brass vessel. Specific heat is 0.1 calorie per gram per degree Celsius of mass 100 gram. The resulting temperature is, let's start. Please understand the problem. They are saying we have got two things. You have got 20 gram of boiling water, right? 20 gram of boiling water. That means water is at 100 degrees Celsius, and you are mixing it with ice cold. Ice cold basically means 0 degree Celsius brass vessel of mass 100 gram. 100 gram of brass vessel, right? And when you are going to mix it, let's assume that their common temperature is T. I mean the temperature of mixture is T. Mixture temperature is T. Got it? So this is cooling down, this is getting heated up. So we are going to simply say they are also saying that there is no heat loss. They said this? No, but we are going to assume it, है ना? So we are going to say the heat M1 S1 delta T1 is equals to M2 S2 into delta T2. So let's talk about water first. So water is 20 gram. 20 gram, let's keep it in gram. Specific heat is 1 calorie. They are not going to say this, but specific heat of water is 1 calorie. Delta T is 100 minus, let's say temperature of mixture is T. Mass of brass was 100 gram. Specific heat capacity is 0.1 and change in temperature is going to be T minus 0. Is going to be T minus 0. Doston, this point can be cancelled out by this 0. This will be written as 20 multiplied by, I can also cancel out this, by the way, right? So I'll get 200 minus 2T is equals to, doston, this everything gets cancels out. I've got T over here. That means 3T is equals to 200. That means T will be equals to 200 by 3. Dosto, 100 by 3 is 33.33. Just multiply 2 with it. That means it will be 66.66 degrees Celsius. 66.66 degrees Celsius. Option first is correct. Clear? Everything is good. Over here we are going to using the concept of law of mixture, right? We are assuming that there will be no heat loss. So if this is heated up and this is cooling down, heat exchange will take place and there will be no heat loss, and this is your answer. Quickly let me know in the chat box if everything's clear, dosto. Be quick. If you have any doubt, poll kahan se milega, you can attend poll from YouTube chat only. है ना? If you are going to ask a single doubt, देखो, a single student asked me about velocity time graph. I went back, I draw the velocity time graph for that student. Okay? So ask me your doubt, I'm going to explain. Now let's start. The first diffraction minima due to single slit diffraction is seen at 30 degree. For a light of wavelength 5000 angstrom, falling perpendicular on the slit, the width of the slit is, let's start. Please remember, for diffraction, you are going to use a different formula that you have learned in YDAC, right? You are going to inverse those formulas. So, first diffraction minima. So, first diffraction minima, by the way, dosto, we actually write delta x as a sine theta, where a is the width of the slit, and that is equals to n lambda for minima. For minima. Please remember, this was the formula for maxima in YDAC. Over here, we are going to use it for minima. This is so simple, right? Now they are saying that angle is 30 degrees. So sine 30 degree is equals to first minima means n is equals to 1, and lambda they have given it is 5000 angstrom. So 5000 multiplied by 10 to the power minus 10. So A will be equals to, doston, this is 1 upon 2. Hopefully I can write 2 multiplied by, no problem. I'm not going to save steps for you. A times 1 upon 2, this is 5000 times 10 to the power minus 10. And now you can find out A can be written as 5000 times 2 multiplied by 10 to the power minus 10. So, dosto, if you are going to pay attention, this is nothing but 10,000, right? This is 10,000 into 10 to the power minus 10. Now let's see how they are giving answers. They are giving answers in centimeter. So let's also write 100 and let's write centimeter, because right now the answer was in meters. Doston, 4 zeros and 6 zeros. So 6 zeros we are going to cancel out, 10 to the power minus 4 centimeter. 10 to the power minus 4 centimeter, or you can rewrite this thing as 10 times 10 to the power minus 5 centimeter. Option 4 is absolutely correct. My dear friends, you might have done some mistake. B is not correct, but I'll check my answer again. You were saying it's going to be B. Let's check it again. D is the correct answer according to me. If you have any doubt, do let me know. Clear? Can we move on? Done? Moving on to the next question. Next problem. See, in this last time, sir, why not option B? Fine, I'll explain. But first tell me why option B here. If we are getting option D, then why option B? That's my response. Right? Sir, please give me more sums on calorimetry. I've got more sums on calorimetry. Please watch the entire session. I've got more. Sir, why not option B? Bache, because option D is correct. That's why not option B, hai na? Why not option D? You tell me. See, n lambda sin 30 is 1 by 2. 2 times 5,000 is 10,000. This is 10 to the power minus 6, and convert it into centimeter, because they are asking answers in centimeter. That's why. Okay? Everything is correct here. Chalo, moving on to the next question. If you have any doubt, ask that. 1 mole of an ideal gas at temperature T1 expands according to the law P upon V is constant. P upon V is constant. Find the work done when the final temperature becomes T2. Start. Start, my dear friends. For work done, you have to use formula. Acha, before talking about work done, let me tell you, this is P upon V. That means I can rewrite this thing as PV raised to the power minus 1 is equals to constant. Is that correct? So isotropic constant becomes minus 1. We actually write it as X, right? So it becomes minus 1. Done? It becomes minus 1. Now, my dear friends, work done formula is NR del T 1 minus gamma. And this is not gamma, this is X. So we are going to get NR T2 minus T1, and I think a number of moles is 1 mole. So 1 multiplied by R divided by 1 minus of minus 1. That means answer will be R times of T2 minus T1 divided by 2. Clear? Done? So R by 2, T2 minus T1. Please remember this thing. The general formula is, the general equation is for an isotropic is PV raised to the power X is equals to constant. And if this is the case, this is going to be the work done. Do remember this. Right? And over here, this is NR delta T. You can use it for adiabatic also. NR delta T divided by 1 minus gamma. Alright. Moving on, option second is absolutely correct. Very, very good. Hopefully, those your doubt from this one has got cleared. This is another nice problem. Read it carefully and then solve this. Let's start. 60 seconds. They are saying a body executes SHM under the influence of one force and has a period T1 seconds, and the same body executes SHM T2 seconds when under influence of another force. When both forces act simultaneously, what will happen? Dosto, the thing is, let's suppose force F, we are talking about force F1. So F1 can be written as K1 times X, right? K1 times X, and hopefully all of you remember that we can write down omega as under root K by M. I'm going to write this over here. Omega can be written as under root K by M. Correct? K over here is SHM constant. है ना? Same K in wave will become wave number. Right now it is SHM constant. Omega is angular frequency, M is mass. Done, bachche? So we all know कि sir time period can be written as 2 pi under root m by k. M by k. So m by k1. We can write it like this. Hopefully this is all clear, right? Now what I'm trying to say is, and similarly T2 will be written as 2 pi under root m by k2. But when both the forces are going to start acting together, you are going to write down T equivalent as 2 pi under root m upon k1 plus k2, that is k equivalent. So I have written over here this is the formula for k equivalent, that is k1 plus k2. Now I want you to use that formula dosto. Okay, pay attention, I want you to use this formula because k can be written as omega square times m. Omega square times m and omega square can be written as 4 pi square divided by t square times m. So that means this will be written as, k equivalent by the way will be written as 4 pi square t equivalent square, and this will be written as 4 pi square divided by, and I can cancel out mass by the way, so I don't have to worry about mass, but still I'll write it for you, divided by k1 square plus 4 pi square m divided by k2 square, and instead of k2 over here I'm going to write t. So t1 square and t2 square. So this will get cancelled out and you will get your answers of t equivalent from here. Pay attention dosto. So 1 upon square of t equivalent will be, this is 1 upon t1 square, 1 upon t2 square. So this will become t1 square plus t2 square divided by t1 square and t2 square. If you are going to reciprocal it and write down t equivalent, this will become t1 t2 divided by under root of t1 square plus t2 square. So t1 t2 under root of t1 square plus t2 square, option 4th is absolutely correct. Clear my dear friends? Quickly write down in the chat box if this concept is clear. 4th is absolutely correct. Very good. Do remember this, है ना? Do remember this. If you are going to do one of these things, you are going to use this in at least 4 or 5 chapters. SHM, wave on string, sound waves, alternating current, wave optics. You are going to use this many times. Time period is often used in many places. So do remember this. Alright, moving on to the next question. Another nice problem from fluid. Question is saying if the terminal speed of the sphere of gold is 0.2 meter per second in a viscous liquid. See, they have also given us density of gold and density of fluid. Find the terminal speed of a sphere of silver. Density is given of the same size, radius is same in the same liquid. Your time begins now. Try to solve it under 60 seconds. I'm going to help you with the formula. Dosto, terminal velocity is 2 by 9 eta in the denominator, r square g multiplied by density of solid minus density of liquid or fluid. Density of liquid or fluid, right? Remember this formula, very, very important. Done? Clear? Now they are saying velocity of gold is 0.2. Terminal velocity of gold is 0.2 meter per second is equals to 2. Let's say radius is r, eta is, let's say, eta, and times g. Density of solid is 19.5 times 10 to the power 3, and density of liquid, that is fluid, is 1.5 into 10 to the power 3. Hopefully this makes complete sense. Formula is good for you. 19.5 minus 1.5, this will become 19 and 18. So this will be, dosto, if you are going to solve this, pay attention. This will become 0.2 r square g divided by 9 times eta, and this will become, dosto, 18 times 10 to the power 3. 18 times 10 to the power 3, right? Now pay attention. We don't have to do anything. We have to find out the value of this. Okay? And how you are going to do this? See. You can cancel out, if you want, you can cancel out this 2, but what I want is you can write down 2r square g divided by 9 eta equals to 0.2 divided by 18 times 10 to the power minus 3. Let's keep it like this. Let's keep it like this. Next thing is, let's apply the same thing for the silver. So for silver, dosto, terminal velocity will be written as, for silver, 2 by 9 eta r square g, and as you guys can see, I already know that formula, I know that value, times density of silver, that is 10.5 times 10 to the power 3 minus dosto density of liquid is 1.5 into 10 to the power 3 is equals to, so this value is equals to that, which is 0.2 into 10 to the power minus 3 divided by 18 multiplied by, this is 5 gets cancels out, this is 9 times 10 to the power 3. That's why I said that do not try to solve that. 10 to the power minus 3 gets cancels out by 10 to the power 3. This becomes 2. This becomes 0.1. So 0.1 meter per second. Answer 3 is absolutely correct. Good. Very good. Time for your doubts. Please let me know in the chat box. Option A is absolutely wrong. Option C is correct. Okay? Option C is correct, bachche. Let me know if you have any doubt. Done? Clear? Sorted? Can we move on to the next one? Please remember this formula. This is very important. Tomorrow, you might get a question from terminal velocity. It might be related with units and dimension, or it might be something like this, or it might be because of those viscous force and finding out the terminal velocity when a body is free falling in a fluid, right? Moving on to the next question. Another very nice problem. This will also clear your multiple doubts. All right. ठीक है, just I'm explaining you if you are worried about tomorrow's exam. ठीक है. I'm explaining something to you. Don't worry. First, let's solve this question. Then at the end, I'll speak with you for 5 minutes. Okay. First, let's solve these questions. Time is on. They are saying the transverse wave is traveling from left to right. So this is the direction of wave. This is the direction of wave. So this is wave velocity. This is wave velocity. VW, not V omega, VW. Then they are saying the adjoining figure represents the shape of the string at a given instant. At this instant, among the following, choose the wrong. See how important it is to actually understand they are talking about wrong statement. Else, you will think they are talking about correct statement. You will select the first one and you will move on to the next question. So please, for tomorrow's paper, do remember this, whether they are talking about correct, incorrect, wrong or false, true, whatever they are saying, read it twice. Dosto, the concept says the velocity of particle is negative times of slope multiplied by wave velocity. Please remember this, multiplied by wave velocity. This is the concept. This is the only concept you have to use. Now pay attention. Pay attention, dosto. All of these points A, B and C, they have got positive slopes. A, B, C, they have got positive slope. Right? Positive slope. That means all of these particles are actually coming down. All of these particles are coming down. And actually the more slope is, this has got the minimum slope, so minimum velocity. More slope, more velocity. More slope, more velocity. And positive slope coming down, negative slope going up, negative slope going up. So these are moving down. First statement was point D, E, F have upward velocity. Points D, E, F indeed have upward velocity because they have negative slope. Correct statement, correct as they have negative slope, as they have negative slope दोस्तों. Correct, correct. Negative slope. If negative slope, then positive velocity. If positive slope, negative velocity. That is the formula. Just remember it for today. Tomorrow, if you are going to get a question, you will easily do it. Now, point A, B, H have downward negative velocity. A, B. A, B is correct. Where is H? H is also correct. Ki sir, H has actually positive slope, so it will have a downward velocity. Absolutely. Please don't do any mistake. H has a positive slope, right? So this statement is also absolutely correct. Point C and G have zero velocity. Yes, yes, you might be saying ki sir, you have said that it is moving down. But just now I said ki this will have minimum slope. Minimum slope, and minimum slope is zero. So it is actually at rest. This particle has slope equals to 0. As you guys can see, so this is at rest right now. So G is also at rest. So this statement is also very correct, which means D is going to be our answer. But let me read it for you. Point A and E will have minimum velocity. I have said that velocity depends upon slope. More slope, more magnitude. Bhaiya, E and A and E have maximum velocity. They have got maximum velocity because they have got maximum slope. So this is incorrect statement. This is incorrect statement. Option number 4. Whew. Only 28.57 were able to solve this, right? So kindly see, this is a very nice problem. And if you are going to only remember this small thing for tomorrow, a big topic will get over for you, right? Moving on to the next one. If you have any doubt, do let me know. This is the next problem, dosto. I've got questions from almost every chapter from the syllabus. Stress required in a wire produced 0.1% of strain. 0.1% of strain and is 4 into 10 to the power minus 8 Newton per meter squares. Its Young modulus is Y1. If stress required in another wire to produce 0.3% strain is this, its Young modulus is Y2, then you have to find out the relation between Y1 and Y2. Come on, be quick. Find the relation between Y1 and Y2. Find the relation between Y1 and Y2. Y1 is stress, and stress is, by the way, 4 into 10 to the power 8 divided by strain. My dear friends, when somebody gives you strain as 0.1%, na, so you can write it as 0.1 divided by 100. Got it? This is 0.1 percentage. So let's write 0.1 percentage over here. So 0.1 multiplied by 100. So this will become 1000. So this will become Y1, you will get 4 into 10 to the power 11 Newton per meter square. Everything is in SI unit. Strain obviously doesn't have any unit, Newton per meter square, and Y2 will be maximum stress is 6 into 10 to the power 8 divided by, now the strain is 0.3, that means 0.3 divided by 100. I'll write 100 over here, one more 0, this becomes 2, this becomes 2 times 10 to the power 11 Newton per meter square, which means that दोस्तों Y1 can be written as 2 times of Y2. Y1 can be written as 2 times of Y2. Y1 can be written as 2 times of Y2. Option B. Clear? Good. Very, very good. Great. Moving on to the next question, dosto. Two resistors of resistances R1 equals to 300 plus minus 3 ohm, R2 equals to 500 plus minus 4 ohm are connected in series. Chalo, this is very easy, starting 60 seconds. Till then, let me see if you have any doubt. Be quick. Clear? This is very easy, dosto. R1 plus R2. They are talking about R equivalent. First of all, R equivalent will be, because it is in series combination, it will be 300 plus 500, and errors are always going to add. Error will always going to add, that means R1 plus, delta R1 plus delta R2. No matter what happens, they are going to always add up, that means 3 plus 4 is 7 ohm. So answer will be 800 plus minus 7 ohm. 800 plus minus 7 ohm, option B. Good. Very, very good. Moving on to the next one. So this is slightly better. Please do it correctly. Okay? Don't do any mistake. Let's start. 60 seconds. You have to find out the displacement of the body in last 2 seconds. They are saying initially the body was at rest. Initially the body was at rest, moving with uniform acceleration A. Its velocity after n seconds is V. Its velocity after n seconds is V. Let's start. In last 2 seconds. In last 2 seconds. Be quick. Be really, really quick, दोस्तों. In these types of problems, you can actually imagine something like this. Let's suppose it is starting from this point, from t equals to 0. Its velocity is 0. And let's say this is t equals to n seconds. Right? And they are saying its velocity after n seconds is v. So first of all, we can find out the value of acceleration. Acceleration will be simply v minus u divided by t. That is v by n. So acceleration is v by n. Acceleration is v by n. Right? Next is they are saying in last 2 seconds. So let's suppose this is n minus 2. This is n minus 2. Correct? So what we can actually do is we can find out the total displacement. We can find out the total displacement, and we can find out the displacement till here. That is s n minus 2. And then you can subtract it. So s n minus 2th second will be Sn minus Sn minus 2. Done. And for this, you are going to use ut. U is 0. So half a into t square minus, again for this one, you are going to write half a into t square. Done. Let's solve this. 1 upon 2. Acceleration is v upon n times n square. One of the n will get cancelled out. Minus 1 upon 2 v by n. And this will become n minus 1 whole square. So if you guys want, you can solve this. Right? And if not, then please don't solve this. N square plus 1 minus 2n. Done. And solve this. I'm going to give you answer. You only have to, this will be 2v n minus 1, 2v n minus 1 divided by n. If you're going to solve this, you will get this. 2v n minus 1 divided by n. Divided by n. Option A is going to be absolutely correct. Very good. Solve this. You hopefully everybody can solve this equation. Okay? Chalo, moving on to the next one. Next is this. Which one of the following represents displacement time graph of the two objects moving with zero relative velocity? All right. Zero relative velocity. Let's start. For the first two graphs, actually both of them are rest. Both are at rest. Because displacement is not changing with time. Displacement is not changing with time. Right? Displacement is not changing with time, so this is at rest. Whereas for this one, this has slope is positive. Slope is a positive constant, and what I can see is whatever slope this graph has, same slope B also has. So this angle and this angle are same. So slope is positive and constant and same slope. So same slope basically means same velocity. And same velocity basically means zero relative velocity. So option B is absolutely correct. As you can see over here, we have got different slopes. And for this one also, B is at rest and A is moving with constant velocity. So answer is going to be second. Answer is going to be second. Please see. When they are saying they are talking about moving, है ना? So the problem with this is that you have not read the question properly. They have said that moving object should be moving. So this was not moving. Arey bachche, this was not moving, na? This was not at all moving. They are saying moving with zero relative velocity. Clear? That's why option first is not correct. Hopefully this makes sense. Clear? Yes, nice problem. Moving on. Sir, please explain again. But I have explained it. Dost, see how much I have written. They both are at rest. Okay? They both are at rest. So even if their relative velocity is zero, but because they both are at rest, we can't choose this answer. They have said for two objects A and B moving with zero velocity, zero relative velocity. Right? So they have, and how to find out velocity? By finding out the slope of displacement time graph. So these two are parallel lines, right? So that means slope are same slope, same velocity, relative velocity zero. These have different velocities. Chalo, moving on to the next question. Let's start this. You have to find out the center of mass of this system. Center of mass of this system. Check calculation of previous one. Beta, calculation of previous one is absolutely correct. If you are talking about that acceleration wala case, that is absolutely correct. No doubt in that. No doubt. Pranjal, that is absolutely correct. 100%. Be quick. Let's find out the center of mass of three particles. Center of mass of three particles. Three particles of mass is 1 kg, 3 kg and 2 kg. Please see. The coordinates of this will become a comma 0. Coordinates of this will become a comma 0. Coordinates of this will become, dosto, kindly note, this is a, this angle is 60 degree. This angle is 60 degree. So this length will become a cos 60 degree. This will become a sin 60 degree. So coordinates are going to be a cos 60 degrees, a root 3 by 2. A root 3 by 2, comma, this height is a by, this is a by 2. This is a by 2. This is a by 2. Right, of course. And this is a root 3 by 2. Done. And coordinates of this is 0 comma 0. So now find out the XCOM. XCOM will be M1 X1 1 into 0 plus M2 X2, that is 3 by 2 kg times X2 plus M3 X3. So 2 into A by 2 whole divided by 1 plus 2 plus this. So that means 3 plus 3 by 2. 3 plus 3 by 2. Dosto, if you are going to pay attention, this will become A, and finally you will get 3 by 2. Acha, let's not cancel it. Hai na? Let's not cancel it. And this, we have got 3 over here. This will become 5 A by 2 and I can write down 2 over here, 2 over here. So this will become 9 and 2 over here. So first answer is 5a by 9. So either it's going to be 1 or 3. Fourth can't be the answer, second can't be the answer. Okay. Next is YCOM. YCOM will be, first one will be obviously 0. Second one will also be 0 because this is 3 by 2 and this is 0. And plus third one is 2 times of a root 3 by 2 divided by, you have got 3 plus 3 by 2. So 2 times 2, and for YCOM I'm going to write it over here for you. Pay attention. Doston, this is 0, this is 0, this gets cancels out. This is a root 3 divided by, we have got 9 over here and you have got 2 over here. Doston, this 9 can be written as 3 times 3. This root can be cancels out over here. 2a divided by 3 root 3. 2a divided by 3 root 3. Option A is absolutely correct. Right? Option A is absolutely correct. Please check. Please check this answer, dosto. Clear? Pranjal, about that last problem, my dear friends, everything is sorted. This is absolutely correct. No doubt about this. YCOM is 2a by 3 root 3. Only 17.86% were able to solve this. Done? Any doubt? Any doubt? Can we move on to the next question? Please see. Please see. A U tube contains water and methylated spirit separated by mercury. The mercury columns in the two arms at the same level with 10 centimeter of water in one arm and 12.5 centimeter of spirit in the other, as shown in figure. The relative density. Find out the relative density of the spirit. Be quick. You have to find out the relative density of the spirit. Clear? Very good. Use the concept that at same level pressure will be same. So this and this level is exactly the same. So you are going to simply say that pressure from here is going to be P naught plus density of water G into H, and H is 10 centimeter. Let's keep it in centimeter, is equals to pressure from here is going to be P naught atmospheric pressure plus density of spirit times G into 12.5. So P naught can be cancelled out. After cancelling this out, G can be cancelled out, and rho S divided by rho W will be, rho S divided by rho W will be 10 divided by 12.5. That means 100 divided by 125. That means 4 divided by 5. That means 0.8. 0.8 is the correct answer. B. B is the correct answer. All right, clear? Done? Any doubt? Look at over here. Moving on to the next question, dosto. Hopefully everything is sorted. Find the combination of, find the equivalent capacitance of this combination. Let's start. 60 seconds. Begin. Begin, begin, begin. Be quick. Find the equivalent capacitance of this combination. Trick is, first find out the capacitances which are connected between same points. Like for example, as whatever this point is, let's say P, this will be P, this will be P, and let's say this is Q, this will be Q, and this will be Q. That means combination of these three are going to become 3C. So instead of three different capacitances, we can write 3C. Now this 3C and this 3C are in series combination. Are in series combination. So this will become 3C by 2. Got it? This will become 3C by 2. I am going to also draw the diagram for you. So I have got now 3C by 2, right? 3C by 2, and then I have got another capacitance over here of 3C by 2. Of 3C by 2. And then you have got 3C, and next thing is you have got, you have got another capacitor over here of 3C by 2. So dosto 3C by 2, 3C by 2 can be actually, now this thing can be removed and we'll have 3C over here. And again you have got 3C. So 3C and 3C, they will be removed, and we can, instead of these two, we can write 3C by 2. And finally 3C by 2 and 3C by 2 can be written as 3C. So 3C is going to be the correct answer. B. 3C is the correct answer, bacha. Only 45% were able to do this. Quickly write down in the chat box. B is absolutely correct. Clear? Any more doubt? Done. Moving on to the next question. Reading of the ammeter will be. Find the reading of the ammeter, dosto. 60 seconds, let's start. Please note this, that these two resistances are connected between same points. Let's say if this is P, this is also P. And these two resistances are connected between same points. If this is Q, this is also Q. And this looks like a balanced Wheatstone bridge. This looks like a balanced Wheatstone bridge because 6 upon 1 is indeed equals to 6 upon 1. So you can remove this 6 ohm. So if you are going to remove this 6 ohm, now you will have, this will be in, this is going to be 12 ohm. This is going to be 12 ohm. So R equivalent will be 6 ohm. 12 ohm and 12 ohm are in parallel, so R equivalent is in 6 ohm. Current will be 2 divided by 6, that is 1 by 3. 1 by 3 ampere. 1 by 3 ampere is 0.33. 1 by 3 ampere is 0.33, that is option 3. Alright. Done? Clear? Any other doubt from here? Be quick. Find the charge stored on the capacitor in steady state condition. Find the charge stored on the capacitor in steady state condition. You have to find the charge on 3 microfarad. Let's start. 60 seconds. Be quick. Remember, why not option B for the previous one? Bache, okay, after this question I'll go back. Okay? After this question I'll go. For the previous question, option B was the correct here. Acha nahi, for the previous one, option B was 3.3. By 1 by 3 is 0.33. That's why. Please remember that steady state basically means that capacitor will behave as an air gap in steady state. Okay? That means T equals to infinity, and it behaves as a clean conducting wire at T equals to 0. So what's going to happen is simply remove this circuit. For now on, simply remove this circuit. Removed. So if this is being removed, this will become 2 and 2, this will become 1 ohm. 1 plus 4, that means R equivalent will become 1 plus 4 ohm, that is 5 ohm. So current will become voltage, that is 10 divided by 5, that is 2 ampere. Now current is 2 ampere, hold on. That means when this 2 ampere will pass through this resistor, there will be a potential drop of, 8 volt over here. There will be a potential drop of 8 volt over here, which means that 8 volt will be over here also at steady state. So if you have got 8 volt, that means charge will be equals to C times V, and C is 3, I believe, 3 microfarad times 8, that is 24 microcoulomb of charge will be there in steady state. First is absolutely correct. Nice problems. Again and again you guys are messing up with options. Only 3 students were able to solve this. Please note 24 microcoulomb is absolutely correct, I believe. If you have any doubt, you can ask me. Clear? A is absolutely correct. Sir, answer A. Yes, answer A is absolutely correct. If you have any doubt, do let me know. I'll repeat it again because only I think 15% were able to solve this. Hold on. Whenever they are going to talk about capacitor in steady state, please remember that we'll actually apply air gap around across capacitor, and we'll remove that branch. If you're going to remove this branch, now the circuit looks like 2 and 2 are in parallel. That means 1 plus 4 is 5. I comes out to be 10 divided by 5. Clear? 10 divided by 5 is 2 ampere. Now please remember, when this 2 ampere will cross this 4 ohm, there will be an 8 volt of potential drop over here, and similarly 8 volt will be on 3 microfarad on this. That's why the current is not moving. Got it? That's why the current is not moving across that arm. Pranjal, just now I've repeated it, bache. Hopefully now it is clear. How is it 8? How is it 8? Because those 2 times 4 is 8. And then we are saying, please understand what we are saying. If at steady state in this arm current is 0, how's that possible? How's that possible? That is possible when it gets fully charged. When it gets fully charged. See, understand this like this. Understand this statement. Let's suppose this is V. So this is going to be V minus 8. Got it? This is going to be V minus 8. Done. There will be a potential drop on this. Now what I'm trying to say is if entirely this 8 volt will be given to this, then current will not flow further because this is fully charged. Done. Can I move on to the next question? Next one. All right. Next question. Two dielectric slab of dielectric constant K1 and K2 of same thickness is inserted in parallel plates, and K1 is equals to 2K2. Potential difference across slabs V1 and V2 respectively. Then let's start. 60 seconds. 60 seconds. Quick. Done. Quick, quick, quick. They are saying K1 is equals to 2K2, right? Let's suppose K2 is equals to K. So K1 will be 2 times of K. Now we know potential, when we write down potential V2, we are going to write it as V divided by K. When you are going to write down potential across this dielectric, you are going to write V1 is equals to V divided by K1, and that is V divided by 2K. That's it here, है ना? So if you are going to multiply, if you want to find out V2, simply multiply 2V1. Multiply 2 with V1. So 2V1 is equals to V2. 2V1 is equals to V2. Option C. Good. Very good. Clear? Done. Potential becomes 1 upon k times. Potential becomes 1 upon k times. Done, done, done. A tuning fork of unknown frequency makes 3 beats per second with a standing fork of frequency 384 Hz. The beat frequency decreases when a small piece of wax is put on a prong. Okay, let's start. 60 seconds. A tuning fork. So they have said we have got a tuning fork of unknown frequency and it makes 3 beats per second with a standard frequency of 384. So 384 Hz and then beat frequency is 3 beats per second. That means either it's going to be 384 plus 3, that means 387, or it's going to be 384 minus 3, right? 384 minus 3. Done. That means 381 Hertz. Here 381 Hertz. Now the thing is, they are talking about the beat frequency decreases when a small piece is put on the prong of the first means tuning fork of unknown. So, and then beat frequency is decreasing. So if beat frequency is decreasing, it has to be 387. I'll tell you why. Because when you are going to apply wax on it, this will decrease. And when this will decrease, the difference between delta F is going to decrease. Rather, in this case, if you are going to apply the wax, this frequency will decrease and the difference between them will further increase. So it has to be 387. That is option 1. Bhai, if you are going to apply wax on this, है ना, then let's suppose this becomes 379. So what is the beat frequency now? 5 beats. So it is increasing. They are saying that the beat frequency actually decreases. So option is going to be 1. Option is going to be 1. Wrong, wrong. All of you are wrong. Bhai, 3 beats per second basically means either it's going to be 381 or 387. Now they are saying the beat frequency decreases when small piece of wax on the prong is first applied. Just a second. Just a second. So frequency will decrease, na bachche? Frequency will decrease. Clear? Any doubt? Any doubt in this one? Chalo. If you have any doubt, do let me know from the last question. है ना? If you are going to add mass, frequency will be decreased. Clear? Clear, clear, clear. Decreased in a frequency. Absolutely. Quickly let me know in the chat box. Chalo. Next problem, poll night, 60 seconds. Let's start. Let's start. Nahi bachche, that's not what they were saying. That's not what they were saying. They were saying ki after waxing, the beat frequency is decreasing. So that means the difference between the frequency of the prong and the another prong, whatever that tuning fork is, that is decreasing. So when you are going to apply wax, its frequency will decrease. That's what the question is. ठीक है? All right. In these types of problems, we have got a tricks. You can directly apply Snell's law between region 1 and region 4. You can directly apply the Snell's law between region 1 and region 4. That means n naught sine theta is equals to n naught by 8, and then they are saying the angle of incidence theta for which it just misses the region 4 is sine 90. You can directly apply this. So theta will be sine inverse of 1 by 8. B is absolutely correct. Good. Please see the next previous question again. They were saying the beat frequency decreases. They are not saying, they are not talking about frequencies of individual things. That you have to think about it again. Okay, that you have to think about it again. If you will wax it, what will happen? If you are going to wax this, if you are going to wax this frequency is going to decrease, and if you are going to wax this frequency will decrease. If this will decrease, the difference between them will increase. If this will decrease, the difference between them will decrease. They were talking about beat frequency decreases. Chalo. So with this, my dear friends, now let's solve 1 or 2 questions on logic gates also, and then we are going to continue from 8 p.m. 8 p.m. you have got another lecture, physics lecture, right? So at 8 p.m. that's going to be last revision for your RE-NEET exam. So late night revision, I'm going to bring the most important topics only at 8 p.m. This is the last problem, then we are going to move on. We'll conclude the session. Dosto, over here you have got, this is output, this is, these twos are input, and we have got output over here, right? So what you can do is, you can draw a truth table. This is A, B and Y. So now A is 1. See, A is high, 1, B is high, 1 and 1, and output is 0. Output is 0. So it can't be OR gate. It can't be OR gate, hai na? Done. Then 0 and 0, 0 and 0, and this is 1. So it can't be AND gate. It can't be AND gate. Clear? Next is 0 and 1. So 0 and 1, and this is 1, and this is 1. So 0 and 1 is going to be 1 for OR and for NAND. So hold on. Then 1 and 0. So 1 and 0. So 1 and 0 is 1. 1 and 0 is 1. So dosto, it can't be NOR gate also. It's going to be NAND gate. Okay? So it's D. Clear, my dear friends? Quickly let me know in the chat box. Be quick, bhai. Be quick in this all. Sorry, I have not started the poll. D is going to be the correct answer. Done? Any more doubts? You don't have to go further. Clear? Chalo. So now let's wait till 8 p.m. and at 8 p.m. we are going to start our another session. Okay? At 8 p.m. we are going to start our another session. In that session, I'll bring the only important topics for you. Okay? So bye-bye. Thank you so much. Take care and see you at 8 p.m. tonight.